Well, good morning, everyone, and welcome to the Southeastern Interlake Agronomic Update, brought to you by Manitoba Agriculture and Resource Development. In the absence of being able to have in-person meetings, given COVID-19 realities, this is a way that we're trying to reach out and provide some information. So we're going to get right into it quickly. We've got a lot of speakers on our agenda today. What we did this time around to make things a bit different is we polled various people in the industry and clients, farmer clients, collected together some questions and asked our specialists to respond to those questions. Presentations will be short, 20 minutes each. We're going to go through all of them first. If you have any questions, please enter them in the questions tab, and then we'll respond to your questions after all the presentations have been completed. So we'll begin with our first presenter, Dennis Lang, our provincial pulse crop specialist. Go ahead, Dennis.
Thank you, Terry, and welcome everyone here this morning. We're going to get right into it here with the first question here today. First question is, given the increased demand for field peas in Manitoba and the opening of a new processing plants in Winnipeg and in Portage, is a crop feasible option for growers in eastern Manitoba? Well, I'll elaborate a little bit, but the short answer is be very selective on when it comes to field selection. Lighter soils with good drainage is recommended, and don't chase markets. The temptation in some years to put peas on ground that really isn't suited for peas because the markets are indicating that it could be profitable if you have a good year can be quite disastrous. So keep those things in mind. But let's go into it a little bit here. A slide from a colleague of mine, Mr. Cherry Buss. Good launch is equal to good location. This is not one of those good locations. You can see here it's a very heavy clay soil with poor drainage, and all of a sudden now you get a heavy summer rain and it's not a good launch. So some years, like in 2018, 2019, we had very good conditions for peas in the valley and in eastern Manitoba, but in 2020 we did not. We had lots of rain early on, and that was not good for peas on those heavy soils. So I want to touch on this a little bit here, and I'm just going to show you an example here. This is a picture from Kane, Manitoba. It's not quite in the southeast, of course, but it's an area that typically is a little bit heavier soil, and they've started to grow a few peas in that area. So this is the same variety, planted on the same day, and it's also, the pictures are taken on the same date. So it's actually at the end of July, early August is when these pictures were taken. You can see here by the picture on the left here, my summer student last year, beautiful stand. This field did 80 bushels per acre. Very lighter, a little lighter soil, very well drained. 2 miles down the road, same grower, you can see here, that's not so good. That ended up doing 15 bushels an acre. So can peas work in the southeast? If you have the right fields and you have good drainage and lighter soil, yes. But be prepared that in those years when you get those big summer rains, peas just can't take that kind of rainfall. Okay. Second question I had here today was, if a producer is considering growing an IP or conventional soybean, what changes in agronomy and overall management of the crop would they consider? Well, first thing you got to ask yourself, so what is a conventional soybean or identity preserved soybean? Typically, these are food-grade soybeans. Highland colors are clear to yellow, and they're not tolerant to glyphosate. And that may seem a little, simplistic when I say not tolerant to glyphosate, but you'll understand the reason why in a minute. IP soybeans have desired quality traits such as taste, protein, and oil content desired by the end users. So in that respect, they're bred for those qualities versus bred for yield and bred for things like maturity to the same degree. There are some nice earlier maturing varieties coming along, but overall, or what they're looking for is a desired soybean. Conventional acres last year had about 2% of Manitoba's total acres, and this year we're expected to be up a little bit, mostly just due to markets and growers looking for different options. So we might see 3 to 4% of our acres this year. And these IP soybeans, some of the major markets are Japan, China, Korea, and Malaysia. So the first thing you need to know, I guess, when it comes to growing conventional soybeans is you need to find your buyer first. You need to find who you're going to be selling your beans to and find out what their requirements are for variety selection and what the contract terms are. There are 4 main buyers in Manitoba when it comes to conventional. soybeans. You can see them listed here. Recently, there's been some really good premiums over the traditional glyphosate tolerant or Roundup Ready soybeans. Those premiums have been anywhere in that $2 to $2.50 per bushel range over the crush beans. But it's really important to have that conversation with the buyer, find out what their needs are, what varieties they prefer. These companies all have their preferred varieties they like to work with, and those preferred varieties are based on what their contract sales are like with their end users. That way, if you're growing these varieties, that you'll have good success in trying to market them. The thing that's different this go around with conventional soybeans and what it was in the early 2000s was that in the early 2000s, when we first started growing conventional soybeans, we were taking soybeans that were suited for other areas of Canada or into the US. McCalls, glaciers were a couple of the lines that we pulled from North Dakota, and they would work in the valley, but that's pretty much it. Anywhere else in the province, they wouldn't work. The maturities were just a little bit too long. And now what's happening is these companies that you see here have developed varieties that are suited for our growing region. They have desired quality traits, and they typically will contract with growers their full production and then have that production come back to them in the fall. So that way you always have an outlet for your soybeans. Where can you find some information on the varieties that are currently grown in Manitoba? In Seed Manitoba, we do some variety testing with conventional soybeans. You can see here all the different varieties based on the maturity groups. And we use Prudence as our check. It's been a long-term check. The other thing that's always interesting is that with Prudence, we always use good high-quality seed for our trials. And you can see with some of these newer lines coming in, there are some varieties that look pretty promising. You know, we're seeing 118%, 106% of the check variety. And on this table as well, you'll see that the IDC scores are also listed here as well. So if you're trying to pick a field that maybe has a little bit higher carbonates and salts, you may want to look at this list a little bit more closely and determine which variety would be best for you when you're working with each company. Some of the things that are the same for both the IP soybeans and the glyphosate tolerant soybeans, establishing good plant stands will definitely help with weed control, but it's especially important in conventional beans because you don't have that later season option, if you want to call it that, for Roundup Ready soybeans as you do with, or you don't have the chemical options as you do in Roundup Ready. You have to really be on top of your weed control in conventional soybeans. Again, choosing variety with a good germination, good vigor will definitely help things along. Your target range for plant establishment is very similar, that 140 to 160,000, which means you're planting in that 200 to 210,000 seeds per acre if you're looking at a solid seeded situation. If you're planting row crop beans, typically I'd still be in that 180 to 190 seeds per acre planted. So the moral story here with this is that with conventional beans, getting them off to a good start, good weed control early on is really important. And we'll talk about that in a minute. I made a comment earlier that they're not tolerant to glyphosate, and this is the reason why. This is a field of conventional soybeans, and you can see here I've put a line across it, and you can see a little coloration difference. While this was a field of conventional soybeans right beside a field of Roundup Ready soybeans, and the farmer who was spraying the Roundup Ready soybeans didn't realize there was a difference between them, and we had some major drift in this particular location. And that's something growers really need to be aware of and talk to their neighbors as well. Let them know if you are growing conventional soybeans, let your neighbors know so that that way when they're spraying, they're going to pay a little closer attention to wind direction. And in this case here, it was a very windy day, and you can see it drifted over the majority of the field. Some weed control comments. Again, first thing I mentioned earlier, if you're growing conventional soybeans, let your neighbors know to avoid accidental drift. Weed control and conventional soybeans have a different management package than the herbicide tolerant systems. You really should start with a relatively clean field, avoid fields with persistent weed challenges, and you really should start with a pre-plant incorporator or pre-seed burn off in conventional soybeans, just to keep that field cleaner longer. And then throughout the growing season, definitely want to keep on top of the weed control early on. And the longer you can keep the weeds at bay, the more successful you're going to be. The thing is with conventional soybeans, we talked a little bit earlier about the plant stand, and you have to really pay attention to the seed quality. If you have a lower seed quality or lower germ and you have a reduced stand, so let's say instead of that 140,000 plants per acre, you end up with 80,000 plants per acre. In a herbicide tolerance system, you can keep it clean pretty much through the season. It's much easier. Whereas in a conventional system, if you're down to those lower plant populations, your herbicide options are much more limited, and your timing is also a lot more critical on those. So just making sure you keep your plant population up, making sure that there's no issues with plant stand. A couple of side notes, I guess, when it comes to growing IP soybeans. You want to keep 2 full growing seasons between herbicide tolerant varieties and conventional soybeans in order to avoid the risk of GMO soybean contamination. So that means 2 full growing seasons in between, making sure that that way you'll make sure that there's no volunteer issues. In my experience, some growers tell me, oh, well, soybeans don't volunteer 2 years later. I've seen it happen more often than not, where 2 years after planting, where a grower has, for example, planted peas in this past year and had soybeans 2 years ago, in the peas this year, you could actually find soybeans coming up. And it's a lot more critical in the IP soybeans, because if there is a GMO contamination, that's going to affect what you're going to be able to sell. The buyer may reject it. Corn, volunteer corn is also contamination risk, so avoid putting conventional soybeans on corn ground. One of the last slides I think I have here right now is when it comes to IP soybeans, you always have to keep things in mind. You have to clean your combine to make sure there's no contamination from any glyphosate tolerant soybeans. Clean your augers, belt conveyors, bins, and trucks. There's a protocol in place that most companies have that you're going to be contracting with, and you want to follow that protocol to set yourself up for success. If you don't, then you might end up with some contamination, and that can be a real challenge. If you are growing both the Roundup Ready and conventional soybeans on the farm, you want to harvest your conventional beans first in the fall. That will also help reduce your contamination. But the biggest thing is just making sure that you go through your equipment and clean things before you get into your conventional beans, whether it's the bins or trucks. And also really important to let the employees that are working for you as well know that it's really important to clean these out. If you make sure that your equipment is clean and pulling into a conventional soybean field, that way you'll set yourself up for success on that. So with that, that's the last slide I had. So I'm a couple minutes early here, so I'll turn it back over to Terry.
Thanks, Dennis. I appreciate that. Thank you for sticking within your 20 minutes. Now we'll move on to our next speaker, which is Dane Fraze. We'll have Dane's slides pop up here on the screen. Just a quick question for you, Dane. Do you want me to ask you the questions, or have you integrated that into your presentation?
You're on mute, Dane.
Sorry about that. If you want to ask them, that would be great. Then everybody has a refresher.
Dane,
Sure.
can you go to the display settings right there? Good, and swap out.
Okay.
Swap. Oh,
There, that better?
I have a little bit of a delay. Yeah, that's great. Go. Yep.
Okay, good. Then I'll introduce
Right
our next speaker. Our next speaker is Dane Fraze. Dane is our provincial oilseed specialist. And for Dane, we had 2 questions. Your first question, Dane, now that clubroot has been identified in eastern Manitoba, how concerned should producers be?
Well, yes, you should be concerned, or at least starting to think about it. As agronomists, and ag service providers, it's going to be your responsibility to communicate that message to farmers, because farmers, more often than not, if an issue isn't immediately present on their farm or their field, they don't tend to pay attention, and they may not take proactive measures. So the map you've got in front of you right now shows the 2 RMs in eastern Manitoba, Interlake Eastern, that have been recently identified with positive clubroot cases. The RM of Bifrost Riverton and the center Interlake was identified in 2019, and the RM of Alexander in 2020. And they both had positive cases where galls were found on canola roots. So they've immediately jumped into that red category. Now, what we have here is we've updated our map to be a little bit more harmonized with what Saskatchewan and Alberta are doing, where we just have a quantity of fields identified and a yay or nay on finding the genetic material for clubroot in the field. So a big part of this management strategy for clubroot is reducing the amount of a susceptible host, canola, obviously being grown. So that does mean having a better rotation. So having producers familiar with this map and knowing that clubroot is certainly in your area is going to be an important concern when managing stuff on your farms. Excuse me. So as agronomists, you've likely heard about clubroot for a while, but where to scout and what to look for is going to be the big thing. Especially in an area where it's new and it isn't extremely widespread yet, but it could get there if proactive management isn't taken. So look for areas that are stressed, stunted, prematurely ripening, dying off in low spots, particularly. If we have dry years like we've had for the past 2 to 3, I know the eastern area is a little bit different. You do tend to get more rain. Areas that would normally be considered low spots that are still suffering, even despite a lack of rainfall or that water isn't puddling there and are still dying, dead giveaway that there's something wrong in that area. The top photo there is a depressional area. It certainly looks stressed out. It's more mature than the rest of the surrounding crop, and it's ahead of swath timing at this point even. So now that would be a sure sign to get out of the truck, go into that area, make sure that you have footwear that can be washed, or you're bringing along plastic booties so you're not transferring soil, and dig up those plants in those areas. Don't just pull them out, because if they're advanced in terms of their infection, the clubbed galls on the roots of the plant could just rip off completely and you might not get them out. So it might be a good idea to dig them out just so you can see what's truly happening. And then, I don't know if you can see my mouse at all.
Yeah, we can.
The highlighter, there we go, spotlight, here we go. If you're looking for plants early on in the season, you'll find fairly firm carrot-like texture galls on canola roots. They're firm and hard and white at that point. These galls here were pulled up while the canola was in mid-bloom. So it can happen as early as the 6th leaf stage, but it's most commonly detected about swath timing or slightly before that. So check your field approaches, corners, low spots, drains, anywhere where soil and water can kind of congregate. Those are the ideal spots for clubroot to spread and start developing in an area, as well as areas that have lower pH. Clubroot is favored by acid soils and soils with lower pHs. Doesn't mean that a high pH won't get clubroot, it just means that it's less likely to spread aggressively. But the lower the pH gets, the more aggressive clubroot becomes. Slide froze for a sec. There we go. Now, this photo here on the right came from the Arm of Alexander actually this past summer. It just so happened that the producer had run out of one variety of seed and had to go back to his retailer and pick up another variety, and what was available was a clubroot resistant variety, and ended up seeding right through the midst of a clubroot patch unbeknownst to them, and had a very clear night and day difference of what clubroot resistant genetics would do versus a susceptible host. And you can clearly see on the right hand side that 233P not having any clubroot resistance failed, and that crop died early. It didn't even form seed at this point. So yield losses can be severe, can range anywhere from 5 to 95%. So that is a very good example of what can happen if clubroot is either unchecked or isn't detected early enough. So we had 3 new fields from our disease survey in 2020. The Arm of Alexander and the Arm of Roblin were both new cases. So we're up to 10 municipalities now across the province, with a total number of fields hovering about 45. I have to do the math again to add them up. Now, that's what clubroot is, but how can we manage them, and what is the messaging around managing clubroots for us? Together with the Canola Council, we have developed a message around keeping them low and keeping them local. If you remember nothing else, remember low and local. So in order to keep spores low, things like a proper crop rotation, so a minimum 2-year break between canola crops. So that's canola 1 in every 3 years. I know we can get decent yields by growing canola back to back, but that is an absolute no-no in terms of being a susceptible host to clubroot disease as well as other diseases. Scouting, you can't manage what you don't know is there, so check every canola field every year. Look in those high risk spots around driveways, field approaches, edges, near shelter belts, low spots, kind of that bathtub ring effect, those spots. If you're in a high risk area, growing clubroot resistant hybrids. And if you know you have it on one field in your farm, grow a clubroot resistant hybrid across every field in your farm, and then let your neighbors know as well, because if it's on your field, dollars to donuts, it's on their fields as well. So it's not just a single farm's problem, it's a community thing, just like herbicide resistant kochia, for example. It moves across the landscape very easily, and everybody needs to be aware of it in order to manage it effectively. Controlling host weeds. So we're trying to prevent that green bridge from one year to the next and not allow spores to increase. We've got to keep a lid on spores. So managing volunteer canola, wild mustard, flaxweed, shepherd's purse, those weeds are reasonably easy to control. And in your soybean and your cereal and corn years, make sure you take a hammer to those weeds too and make sure they're dead so that they don't act as a clubroot host in those non-canola years. If the patch is small, you detect early, there's options about using lime, wood ash perhaps, a grassy cover, keeping that area permanently grassed or grassed for a number of years to allow spores to decline. That's an excellent strategy. Once it becomes larger than a patch, then you're looking at a bit more of a comprehensive approach across the farm. But catching it early, patch management, while can be a little inconvenient to set up, is certainly a good way to manage it and reduce spread across an area. So this is really what we're going to do. This is why I said, please don't sue me, Warner Brothers, because this is what I want you to do. Keep a lid on clubroot and keep knocking them down. Don't allow those spores to increase and get to the point where yield infection starts to, or yield loss starts to occur due to infection. So that happens around 80,000 spores per gram of soil, but we'll get to testing in just a second. So that was the keeping them low portion, but how do we keep it local? So keeping spores in a single patch, don't allow them to spread over the field, don't allow them to spread over your farm. Prevent soil introduction to keep clubroot spores out. So if you're having custom machinery come through the farm and do a lot of that work, I request that operator has a clubroot management plan in place. Ask when was the last time that machine was cleaned. If it was a very muddy, very wet spring, doesn't look like we're in for that right now. That certainly can be higher risk in terms of moving mud clods around. So pressure washing and keeping the machinery clean, if you're a custom operator, is going to be important. If you're an ag retailer, recognizing that clubroot can be a concern. If you are dealing with a client that has clubroot and you're doing custom work on those acres, perhaps deal with that entire farm first before moving on to another farm, or at least sanitizing at the end of that field in question, that suspect field. Patch management, I discussed a little bit earlier, but now minimizing soil movement from known clubroot zones. If you only have a 2-3 acre patch, even if it's in the middle or the edge of a field, getting a little bit of grass seed, spreading that down, harrowing it in, and letting it work its way there and survive there for 3 to 5 years is an excellent way of keeping that soil in place and not moving off site, especially if you have lighter soils, sandier soils, peat soils, where that soil can become airborne and move off site a little more easily. Using things like a perennial ryegrass to keep soil in place, an excellent strategy. And then reducing tillage. We are known for heavy tillage in Manitoba, particularly in the eastern side of the province in the Red River Valley. Reducing tillage if possible, minimum till. And if you're going to be tilling, and we know that is a reality, try to do those infected fields last if you know you're going to have an infected spot. And this is where different types of tillage will also make a difference. High-speed vertical till has become very, very popular lately, and it is an excellent residue management and seedbed prep tool. However, because it works so quickly through the soil, you're not moving a lot of soil, but you are moving a small amount of soil a lot further with a vertical till implement than you would, say, with a moldboard plow on the opposite end of the spectrum, where you're moving a huge amount of soil, but it might only be moving a foot or two in terms of that flip. Vertical till could move that soil 10, 12 feet. And if you go through it twice, obviously double that direction. So that's how those patches can become a lot larger if tillage isn't managed properly. Now, I often get asked this question, do we use a resistant variety preemptively? So you can use them preemptively if your individual or regional risk factors are higher. Oh, it's double voicing myself. I think this is a slide that I have pre-recorded some audio. But what I've got here is knowing that clubroot is present in Bifrost Riverton and the Arm of Alexander, that increases the regional risk. If you're farming in those RMs or adjacent to those RMs, certainly start using clubroot resistant varieties. They are really no more expensive than any other hybrids that are out in the market right now, and clubroot resistance in commercially available canola varieties is becoming more and more common, so it's more easy to access those. In Manitoba alone, over 33% of acres across the entire province were clubroot resistant in 2020, and we expect that number to grow. Now, there's differences in terms of resistance. There's first generation, second generation. Some pathotypes are able to overcome first generation. However, when we're in a very new scenario, like we are in Eastern Interlake regions, sticking with a straight up first generation resistance variety should work well. We have no reason to believe that it won't work. So there's no reason to go and panic and buy the most expensive, best genetics in terms of clubroot resistance around. The first generation will work as long as proper stewardship, proper rotation, those things are still practiced. So resistant varieties are not a silver bullet, but they are going to be helping. So where to find more information on that? Like Dennis said, I'm going to promote the Seed Manitoba guide. We've spent a lot of time putting this guide together each year. So we have 50 some odd hybrids that have some form of clubroot resistance as labeled in the seed guide. You can pick up a copy in Manitoba Ag offices. It came out with the cooperator back in early December. And now there's many more Liberty Link and Clearfield hybrids than there have been in past years. They previously were largely restricted to glyphosate tolerant canolas, but that has changed dramatically in the past 2 years. Now, I talked about getting tested for clubroot. If you are in a high risk area or you think the clubroot could be moving into your area, simply sending away a soil sample can give you a good indication if clubroot spores are present in your land and at what concentration. The Manitoba Canola Growers and the Pest Surveillance Initiative, funded by MCGA, allows one free test per grower per year, and I believe they've expanded that program for the past couple of years. So you're able to send in a soil sample, but don't send in the same soil sample you use when you are doing soil nutrient testing. Rather, you want to look at a suspect area around the driveway and those low spots, etc., and just take a cup full of soil right off the surface. You don't need to go down 24 inches. You don't need to aggregate it with soil from across the entire field. If you do that, you're going to run the risk of getting a false negative, meaning that it'll be diluted enough in the soil that you aren't going to be able to pick it up. So just looking at those high risk spots, send a separate sample in from those high risk spots. A cup or two is plenty. Send it into the PSI lab in Winnipeg. Go online to canolagrowers.com to find the contact information and links there. So as an agronomist, you might be offering this service to your clients and being able to have an open discussion and open up the discussion with them. All right. Terry, I think the second question you had was talking about hidden yield robbers. Is that correct?
Yeah, what the question was is generally canola yields were less than anticipated in the area. Was this because of the weather, or are we looking at other potential problems?
There's a couple factors at play here. I did a little bit of research in terms of prepping for this question, and the Eastern Interlake did have a bit more disease pressure than other parts of the province, but I'd like to go through a bit of a diagnostic tool, and then we're going to address that question specifically. So as agronomists, if you're dealing with clients that have poorer than expected yields, poorer than expected crops for whatever reason, this is sort of how my mind map works in terms of determining what could be the problem. I like to start on the left-hand side of the screen there. I'm looking for straight lines, anything that would tip me off that this is not a naturally occurring issue, that it might be a man-made problem, it could be an equipment problem, there could be gaps or doubles in terms of the seed, miscalibration, was it the right implement for the job, the situation faced. If I can rule any of those things out, now I start looking at the soil. Was it too wet? Was it too dry? We might be facing that too dry scenario now this spring, particularly with seed-placed fertilizer injury. That could be an issue. If it was too wet, was trench smearing an issue, compaction, too much residue management, those sorts of things. Then I move on to the environment. Optimum growth conditions, too wet, too dry, temperature being important, especially for herbicide application. What was the temperature at the time it was applied or the preceding 24 hours? Winds for drift often, or even wind burn, wind damage, disease and insects. Then I start looking at inputs. Was the crop choice appropriate? Was it good seed? Did the seed have a high germ? Are we meeting the 4R requirements for fertilizer application? And then lastly, I look at product performance, because usually it's not the product misperforming. I mean, the product, particularly herbicides, have to be registered and proved effective in order to be registered in Canada. So I tend to not think that is the primary cause of concern, but that's often something that a producer might immediately jump to. I like to get to that last. Looking at drift, misapplied rates, misapplied tank mixes, incorrect mixing orders, delayed activity, again, due to weather or some of those previous factors, and then timing. So looking at all these things usually helps me sort out what the problem could be. Now, for canola specifically in Eastern Interlake regions, what was the issue? My colleague David Kaminsky shared this slide with me a little while back. It was showing the trends in disease incidence in canola from 2017 to 2020. The 2020 columns in purple, you'll notice that blackleg has increased a fair bit. It went from 70, 73, 68 to 83% incidence. So 83% of our canola crops across Manitoba had blackleg in them. So that number is higher, and it is somewhat surprising given that we had a drier year. But blackleg is less affected by moisture conditions than, say, sclerotinia, which was significantly down this year. So that's why stem rot being lower where it is. The other increase here, this is probably a mislabeled thing, would be verticillium. David will address that later, but those verticillium numbers do seem to be climbing and increasing, where we have approximately 30% of our crops that we believe are infected. So blackleg being the main culprit of concern when it comes to disease, and looking at the disease severity scale. Oops, pardon me. We want to know where yield losses start to occur. So this nice graphic put together by Canola Council again shows what we're rating for. We have a rating scale from 0 to 5. The average the average severity in Manitoba was about 1.1 to 1.2, so falling in this range. We do tend to see a little bit of yield loss, not terribly dramatic, but if I look at the Eastern Interlake region alone, it was 11 to 12% higher than the provincial average. So on 33% of canola crops in the Eastern Interlake region is where we found blackleg, and that severity increased to about 1.4. So we started seeing more significant yield loss due to the higher severity rating of blackleg in the Eastern region. So that does lead me to believe that blackleg is becoming more and more of a concern, despite rotations being relatively good in Eastern Manitoba. The moisture conditions and the alternate hosts for blackleg making it more severe in that part of the province. So that is one of those hidden yield robbers. It's not easily detected, especially at reasonably low severity levels, because the plant still looks healthy. It's only after swathing, after harvest, where we start seeing those stems start to discolor. Earlier on in the year, we're going to have to be doing stem cross sections, cutting them open, looking at the base of the stem to see what percentage blackleg infection we have happening right there. Blackleg can infect as early as the 2-leaf stage. So things where flea beetle injury comes into play, creates an injury or an opening in that plant tissue, allows the blackleg fungus to infect and start causing disease losses earlier on. Now, this is something that may be becoming more and more common to you as agronomists. You might be dealing with this in terms of your seed sales and making seed recommendations. Farmers are still less familiar with blackleg major gene resistance labeling. It's a real mouthful, and there's a lot of complicated things going on in this slide, but I'm going to try and break it down in a way that's easier to understand, hopefully. Now, When you're buying a bag of canola seed, it comes with a single letter rating, an R, MR, MS, or S, meaning susceptible all the way to resistant. And that's going to give a percentage of infection of the susceptible host West Star. So West Star is not commercially grown anymore, but it is the weakest variety we know of when it comes to blackleg. So in order for a variety to get an R rating, it has to have less than 30% infection of that West Star check. So that doesn't mean that it's going to be completely immune to blackleg, but in a field scale scenario, it should be less than 30% West Star, which usually it is. Normally we're not having 100% of the crop infected, but 30% is fairly common. Now, does that tell me exactly which gene anything is resistant to, or what groups of blackleg are at play? And no, that system has a fault that way. So we're looking at quantitative or field scale adult plant resistance with this sort of system. A number of canola companies, not all, are moving towards a dual system where they're giving this adult plant or quantitative trait resistance rating together with a major gene resistance group label. Now, a major gene is more like a puzzle piece. So for example, I'm going down here on the bottom left, I have to have a blackleg gene that's causing blackleg virus, and it has to match up with the canola gene that's at play. And if they don't match up, I'm not getting that major gene resistance. So that's going to be my big hitter. That's really what's going to take out that specific race of blackleg. If I don't have this combination matching, as I do here in this situation where the blue triangle does not fit into the orange circle, I'm not going to get that major gene. I'm going to have to be relying on my adult plant resistance, which may or may not work depending on the major race of blackleg that's actually in my field. If the blackleg race is not AVR LM2, it's something different, that resistance may not work. But if it does match, for example, if I have in the field, the dominant type is the AVR LM1 blackleg race, and I have RLM1 resistance in my canola plant, they match. That puzzle piece gets put together, and that major gene is going to be doing a great job doing the heavy lifting in terms of blackleg resistance. Now, when they match, those numbers and names are very complicated to remember, and that's really not what we expect a farmer to know. But what we would like them to understand is that equals resistance group A. So just like a herbicide group, you know your group 2s, your group 4s, your group 6s, those are strong broadleaf products. If you have a group 2 resistant weed, you might want to go with a group 4 or group 6 if that is the weed you're targeting. Same thing works in blackleg now. If your resistance group doesn't match what the resistance type is in the field, you have a group A resistant, pardon me, let's change it up. Then you have a group C resistant blackleg gene in the field. Using a group C product or a group C resistance in your canola isn't going to be as effective against that major gene. And that's what we are seeing. We're seeing more and more blackleg because 95% of the canola varieties that are commercially available in Western Canada contain resistance group C. And the gene that's in the blackleg population isn't the corresponding one to RLM3. It's something different. So that resistance group C isn't as strong as we'd like it to be. So those populations are shifting, meaning that we might have to look at different groups or combinations of groups. That's what this two-part label means. It's got a resistance at an adult plant level, but we also have resistance to major groups B and C. But in this case, where RLM1 meets RLM1, then we have resistance group A. So using combinations or having your blackleg stubble tested just to check out what is the major gene might help your clients and farmers pick a better canola variety that would have more appropriate blackleg resistance rather than taking a blind shot in the dark and just hoping that this single resistance label rating is adequate to take care of that disease. So that's one of those major hidden yield robbers that it's a little bit misunderstood and is still coming to the table. So there are a number of canola companies that are using this two-part system. There are others that are sticking with this field resistance label system. And if you're not getting answers out of them, keep pressing them for information as to what that resistance group is, if you know what's in your field. So that same PSI lab through Manitoba Canola Growers does do blackleg race testing, so you can more appropriately match your resistance with what's in the field.
Dane?
Now, yes.
Yeah, I'm going to have to end you here. We have to move on to Anne.
Sounds good. David will talk more about verticillium later, so you'll catch that talk from him.
Right on. Thanks, Dane. So as we're waiting for Anne's presentation to come up on our screen, I'll give it a quick introduction. And Anne, in your case, I'm going to put all your questions out there because it's kind of a group of questions. But the overall theme for Anne is, where are we at with plant growth regulators in cereals? What does Manitoba research tell us about them? Can producers expect an economic return? And are there any updates on the regulatory MRL issues surrounding their use? Whenever you're ready, Anne.
Okay, great, thank you. Okay, so I'll answer those questions, and I do have some updates from PGRs for Canada. So this year, there are 3 PGRs registered for Western Canada. Manipulator is probably the one people are most familiar with. It's been registered since about 2017, I believe. Modus was just registered this December, so this 2021 season is the first time farmers will be able to use Modus. And then Ethyl has been registered for a few years as well, but it isn't marketed in Western Canada just because there are a lot of crop safety concerns. If the timing is very specific for Ethyl, so in order to actually use it, Bayer requires that farmers sign a waiver, and due to that, they're not actually marketing in Western Canada because it's not that popular. So the 2 options people have are Manipulator and Modus. They both function similarly, as they're both gibberellin inhibitors. So gibberellin is a type of hormone that makes plants taller and skinnier. So when that's inhibited in the plant, this will prevent the taller growth. So they do work a little bit differently, but they're both registered for spring and winter wheat, oats, and barley, and have different active ingredients. And I did have the pricing on the side just in case you're interested. There are some different rates proposed for spring wheat, winter wheat, oat, and barley, which is why there's some different pricing depending on the product choice and the crop that you're spraying it on. So I just wanted to go a little bit to answer those questions, I just wanted to go a little bit into the application timing and how PGRs work to get to the answers of those questions. So I'm going to focus on Manipulator and Modus. For both these products, they're registered for a split and a single application, but what we're really recommending is if farmers are going to use them, that they apply as a single application, and the ideal timing is growth stage 30 to 32, which is the beginning of stem elongation. They're both registered until growth stage 39, which is when the collar is visible on the flag leaf, but we wouldn't recommend applying that late. So as I said, the best application would be a single application at growth stage 30 to 32. So prior to growth stage 30, the nodes are still underground, and the nodes really just come up into the plant, into the stem at growth stage 30, and that's when the stem begins to elongate. At growth stage 31, the first node is at least 1 centimeter above the tillering node. In growth stage 32, node 2 is at least 2 centimeters above node 1. So you can see that the timing is very specific, and it's really important to get that right, or so that people aren't wasting money by applying these products. So for Manipulator and Modus, the concern isn't a crop injury. There isn't as much of a concern with crop injury if you apply at the wrong timing. It's just that the product won't be very effective, and therefore it'd be a wasted application. So properly growth staging the plants is important. So pulling plants from a representative area in the plant, and just looking at the main stems, and then using a razor blade to cut the stem in half. So once you cut the stem in half with a razor blade, then you can look at the tillering node, the first node, the second node, and measure those distances. So I just have some pictures here of ideal timing and what would be past ideal timing. So the picture on the left-hand side here would be growth stage 32. So I didn't actually indicate the second node there, but you can see that the tillering node is right at the base of the plant, and that's where the tillers are formed from. The first node is a couple centimeters up, and then the second node is less than 2 centimeters above the first node. So at the beginning of the ideal timing, you will see that first node that's at least 1 centimeter away from the tillering node. And as the plant continues to grow, that node will push further away from the tillering node, and then the second node will develop, pushing its way away from the first node, and then you have the third node develop, and so on. So the picture on the right is past ideal timing. You can see that the plant stem is more developed, and the second node is more than 2 centimeters away from the first node. And you can see that the inflorescence is more developed as well. So I think it's important if someone is considering applying plant growth regulators to be out in the field, you know, when it's close to when you think it's going to be the right timing, just to be pulling some plants and taking a look at them to see when those nodes are starting to appear. So in terms of economics, I think that people are interested in tank mixing plant growth regulators because it obviously saves in that application cost and makes it more cost effective. But it's really not recommended because the ideal PGR application timing is between herbicide and fungicide timing. So while for a plant growth regulator such as manipulator, it's actually registered for all of those timings. So it's registered from about the 2-leaf stage to that flag leaf appearing. It's really not recommended because it won't be as effective. So since manipulator and Modus work by shortening the distance between the nodes, if you spray too early, the nodes aren't there and the product won't work well because it won't last forever in the plant. So at herbicide timing, you know, the nodes aren't present, and at fungicide timing, it's too late because the plant is already elongated and it can't go back in time and shorten the plant after it's already, after the nodes have already elongated. It would just prevent future growth. So in terms of talking about effectiveness, it's important to talk about what plant growth regulators actually do and what you could expect from spraying them. So PGR research in Alberta did find a frequency of height reduction in wheat with manipulator that was 67% and Modus that was 22%. So 67% of the time when manipulator was sprayed on wheat, they did see a height reduction. So that's important to keep in mind that you won't see a height reduction in every circumstance. PGRs aren't like herbicides where you expect them to work the same every time. It depends a lot on the environment, the crop that you're applying them in, and actually the variety of the crop. So the research in Alberta did find that Modus worked better on barley and that they're recommending manipulator for wheat. So in trials that we did at the diversification centers, we didn't see height decreases every time as well, but on average over about 16 site years, we saw 2 to 8 centimeter height decreases on average. And really, I think it's just important to be realistic about what your expectations are for height decreases. There has been an on-farm trial run by the Manitoba Crop Alliance in 2018, 19, and 20. The graph showing here just shows the height decreases that they saw in 2020, and the bars with the stars above them did have significant height decreases. So last year, in 2019 and 2020, they saw height decreases at 77% of the locations. But I think the average height decrease, you know, you wouldn't be expecting huge height decreases. You would just be expecting some moderate height decreases. In terms of yield, it's tough to say with yield because in the years when we studied plant growth regulators at the diversification centers, they did tend to be dry years where we didn't see lodging in the crop, and we wouldn't have, you know, seen any sort of yield benefit from plant growth regulators. On the on-farm trials, I looked at the data from the last 3 years, and there were some years or some locations where they did have, you know, average or above average rainfall at some locations, and in some of those years, they did see a significant height increase or a significant yield increase, and some lodging at some of those sites. So it's really tough to say what you'd expect for yield. It really just depends, you know, what your crop yield potential is and if the crop is expected to lodge. So here from the PGR on-farm trial results last year, they saw one location where there was actually a height or a yield decrease with a plant growth regulator application, and one where there was a 3 bushel per acre increase. And these bars here are just showing the significant yield results. So out of 7 out of 33 site years, there was a significant yield difference between the untreated and the treated with manipulator on the on-farm trials. So of those, 6 of them were significant positive yield increases, and really the average was about 3 bushel per acre yield increase. So plant growth regulators will only be profitable if they reduce the yield losses that might occur from lodging. So they're not intended to increase yield potential in general, and you won't see a yield increase if there is no lodging in the field. They're just there to protect potential yield loss that might occur from lodging. So they're most likely to be profitable when the variety is prone to lodging and when the crop has high yield potential, and when environmental conditions would be conducive for good crop growth, and you might expect some lodging in the future. So it's really tough to say, it's really tough to predict if your crop is going to lodge at the time when you'd have to apply plant growth regulators, and deciding whether or not it's going to be profitable is really would be really difficult. So some things to keep in mind when trying to decide if plant growth regulators are a good fit is that they won't eliminate lodging, but they might delay the onset of lodging. So research has found that in crops that are highly susceptible to lodging, it won't just outright eliminate lodging, but it could delay it from, say, the end of July to mid-August, and that could have benefits for yield and quality. In areas where plant growth regulators have been sprayed and there hasn't been lodging, researchers have found variable effects on yield. So sometimes there's a yield increase due to plant growth regulator applications, sometimes a decrease, and often no impact. It's also important to keep in mind that plant growth regulators are hormones, so they can affect other hormones in the plant, which could result in developmental and physiological changes. So some things that have been seen is the green colors of the foliage could be intensified, which might result in slower maturing crop. Sometimes there's the increased production of late and unproductive tillers or reduced grain weight. It's also really important not to apply plant growth regulators to crops that are under any type of stress. So on all of the labels for these plant growth regulators, that is stated very clearly that there could be negative consequences if plant growth regulators are applied to plants that are under stress. And also researchers in Alberta have sometimes seen a protein reduction after plant growth regulator application. They haven't seen a significant protein reduction, but it is something to keep in mind. So if you don't expect lodging to be a big problem, but protein management is an issue for you, then plant growth regulators likely aren't a good fit. So the other part of the question is plant growth regulators in Europe, the UK, Australia, where they are more commonly used. And the reasons why they're more commonly used, at least in Europe and the UK, is that there is a wetter climate and a longer growing season. So there is more lodging experienced. On average, research has found that there's every 3 or 4 years, there's widespread lodging in the UK, which could occur in up to 20% of the winter wheat area. So if someone expects widespread lodging, and 75% of the years, then I could see where plant growth regulators would become more beneficial. And at least in the 2000s, they'd shown that plant growth regulators were used on 84% of the winter wheat acres in the UK. And I'm not sure what the number is now because I haven't seen any recent data, but yeah, the use is widespread and they do see more of a yield benefit just because they have more lodging. And in some of these crops, the yields would also be considerably higher than ours, just specifically in winter wheat where there's that longer growing season and they do have more moisture. There's more yield to protect because there are higher yields expected. Another thing that's interesting in Europe is that it's common to tank mix manipulator and Modus for improved performance. So while they both work inhibiting gibberellin in the plant, manipulator works earlier in this pathway and Modus later in the pathway. So they have seen some benefits of a tank mix in terms of improved performance and consistency. But right now that's not registered in Canada. There has been some research on tank mixes in Canada with Modus and manipulator, and this research has found that there's more frequent height reductions and a larger height reduction with the tank mix. But that obviously significantly increases the application cost for that product. And in terms of MRLs, I would still recommend that prior to applying a plant growth regulator that farmers do contact, you know, who they expect to sell their seed to, just to make sure that there's no issues with applying a plant growth regulator. But there are MRLs established for both manipulator and Modus. So there shouldn't be too many concerns with those, but I would recommend, yeah, talking to a grain buyer. So just in summary, manipulator and Modus are both registered for wheat, both winter and spring wheat, barley, and oats. So manipulator does seem to be the best choice for wheat, Modus the best choice for barley, and either could work well for oats. And the reaction to plant growth regulators is variety specific. So research done by Sherry Stridehorse in Alberta has identified some varieties that did respond more consistently to plant growth regulators. So if anyone's interested in those specific variety reactions, I could send those to you. And in terms of economics, it's most likely to be profitable when the variety is prone to lodging and when the crop has high yield potential. And when you're targeting those high yields with high nitrogen rates or high seeding rates. So, you know, I think it's really important to think about the crop management prior to applying a plant growth regulator, because it does, you know, add a lot of cost to the overall cost of growing the crop. And that's all I have on plant growth regulators.
Thanks very much, Anne. Greatly appreciated. So next we'll move on to John Gavlowski. So while John's presentation is loading and coming onto the screen, I will read out John's questions. So the questions that we provided to John were: insects continue to be problematic in parts of the southeast and the Interlake. What can we expect in the coming year in regards to grasshoppers, armyworms, and cutworms? And also, what are some of the specific scouting techniques that can be used for cutworms? Go ahead, John.
Okay, you can see my screen, can you, Terry?
Yes,
Or
you're
somebody?
good. Yeah,
I'm
you
good
bet.
to go. Okay. Awesome, thank you. So I'll go right to the questions that Terry had just read out. So these are the two we're going to be answering. What can we expect from grasshoppers, armyworms, and cutworms, and specifically how to scout for cutworms? And just, oh, here we go. So just a recap, last year, armyworms, cutworms, grasshoppers, they were three of what I call our big four insects for the year. The other one would be flea beetles and canola. All four of these insects were at economic levels, at least somewhere in each of our agricultural regions. So they were quite widespread issues across the province last year. And each of them is a little bit different in the way they affect the crop and the way we need to scout for them. But I'm going to start by addressing the second question, scouting for cutworms, how to do that. And a few tips for you here. One, note that cutworms are nocturnal. That means they feed at night, they hide in the day. So if you're out scouting during the daytime, you need to be looking under any debris or stubble or in the soil. Often if you do have a lot of trash or stubble on the soil, they will crawl underneath that debris. They don't want to be exposed. So if there is debris, sometimes just turning over that debris, you will start finding them. If there's not a lot of debris, they will actually burrow right into the soil. So then you need to be digging in the soil. The other thing to be careful of is it's easy to walk into a field like this sunflower field here and start seeing notching on the leaves and no insects around and be really confused. What would have done this? I'm not seeing any insects, but I'm seeing some feeding. There are some species of cutworms that don't do a lot of clipping, so they don't give you what people often use as the telltale signs of cutworms. Some species are more what we call climbing cutworms. They climb, they notch, or they chew, but they don't really clip plants, and that can be very deceiving. And in that case, again, dig around the plants. So if you're seeing plants that have notches, are wilted, dying, do some digging. You can either use a trowel and dig. I usually suggest going down at least 5 centimeters, but it really depends how dry the soil is. The drier the soil, the deeper they're likely going to go. If the soil's moist, they're probably closer to the top. If it's drier, dig a bit deeper. You can try to sift the soil through a sieve if you want, but depends on your soil type. Some of the clay soils, it might be quite frustrating trying to do that. When you're doing your scouting, note the species if you are comfortable doing cutworm species ID. And then the reason I mentioned that, I mentioned some cutworms don't do a lot of clipping. So dingy cutworm, one of our common ones, they're one of the climbing cutworms. They won't do a lot of clipping. They will do a lot of defoliating. Redbacked will. Also, cutworms can grow to a bit different size. Redback gets a little bit bigger than dingy. So redback, about 38 millimeters, so roughly 1.5 inches when fully grown. Dingy cutworm, a little bit smaller, 25 to 32 millimeters, so about 1.25 inches when fully grown. So if you're trying to figure out what stage they're at, are they almost done their cycle? It's good to know what species you're dealing with. If you do start digging up these pupa cases, and you've got quite large caterpillars, you're pretty much done your cycle. They only have they have a one generation a year. They're only larval once. So once most of them are fully grown, starting to pupate, you're done for the season. So that's an important thing to consider as well when you're scouting. And also, are they patchy? Are you able to find them consistently throughout the field, or is it a specific patch in the field? Because with cutworms, when they're laying their eggs, usually in the late summer, when most of the egg laying is done, if there's a good source of nectar somewhere in the field, a weedy patch, a late flowering patch of crop, that might draw them to that area of the field. They lay their eggs. Next year, that's your cutworm patch. And if you can detect that it is patchy, sometimes you don't need to be spraying a whole field. You can be just doing a smaller area. So patch spraying might be an option. And if you do need to treat for them, for cutworms, later is better, just because, again, they're nocturnal insects. Forecasting cutworms, it's a tricky thing to do. There are pheromones available for trap use for some species, but the reality is it doesn't really give us much useful information, so we really don't do it. People tried testing out this technique in the late 80s, early 90s, and there was very poor correlation between trap catches late summer and what happened the next spring. So the traps really, pheromone trapping isn't really used for cutworms. The best thing we can do is tell you when you seem to be in a high cutworm cycle, like we've been for a few years, just plan on keeping an eye on your fields early in the season. Timely scouting is the best advice, especially when we know we've had some higher years. There are a lot of things that can help knock the population back, such as this ground beetle here. You've got parasitoids like bee flies and tachinid flies. If we get a really wet spring, fungal pathogens can sometimes help lower the population as well, but that's a hard thing to rely on. So switching gears to armyworms. And when I use the term armyworm here, I'm not talking Bertha armyworm. I'm talking about a species that tends to prefer grassy plants. Some people will call it cereal armyworm or true armyworm. So this is an insect that migrates in. It doesn't overwinter here like Bertha armyworm and the cutworms do. Some years we can almost spontaneously, it seems, get a high population, and then other years they're barely present. So they're an unpredictable one. Last year they moved in some fairly heavy levels, probably in early June. And when they move in, so this is the adult moth on the left. They're a very light brown moth with a couple white dots on the wings. When they move in big numbers early, they're looking for a dense grassy stand, ideally, to lay their eggs into. So perennial forages, winter cereals are just ideal. And a lot of the fall rye had some heavier levels, and some of the forage grasses in particular. So those are the areas you want to be looking, especially any really early seeded cereals, fall cereals, perennial forages. They're actually in the same family as the cutworms, so they have a lot of cutworm behaviors. They will curl up into a ball when you disturb them. They like to get underneath debris and into the soil during the day. They like to hide during the day when they're older. When they're young, they will feed on the leaves during the day as tiny little caterpillars, but when they get to be older larvae, they are much more like a cutworm, where they like to hide during the day and come up onto the plants at night and feed. and the pupa stage looks very similar to a cutworm as well. And I mentioned they don't overwinter well or at all here. There's been a couple studies done on this. The first one that I'm citing here was actually done in Manitoba, and they concluded that based on their results, the armyworm should not be overwintering on the Canadian prairies. Now, this was done back in the 80s. Climate has changed a bit since, but we still think that they're blowing in. They're not really overwintering here. Similar study in Quebec had the exact same results, that they shouldn't be overwintering in Quebec. And we do know that they have a purposeful migration. People have documented that. They will migrate, so not just being blown by winds, but they actually will migrate north in the spring, and then they do have a southward migration later in the season. You can trap for these, it's possible. Again, they're light moth, and there is a trap called a unifer trap that Quebec uses to try to monitor them, and Ontario some years has a monitoring program going too. Given what happened last year, I have ordered some of these unifer traps, and we will have a network of them across Manitoba this year, probably about 25, 30 traps spread across the province, and I know there will be at least a couple of them up in the eastern region. So we'll keep you posted on our weekly Manitoba crop pest updates, what we're finding in these traps. And if we do see things go from very few to a lot in a given week, we'll give you a heads up, and that will tell you it might be good to start looking in your crop for the larva. On a positive note, this insect does have 2 generations this season, and last year we really didn't see much of the second generation. I think what happened, there's a parasitoid called Cotesia. It's a tiny little wasp, and when they lay their eggs into armyworms, they don't lay just 1 or 2, they lay dozens usually. And the armyworms, they're going to become pretty sick, and before they die, they climb to the top of the plant, and they usually die really high in the canopy. And so when all the wasp larvae emerge from the caterpillar, which happens almost spontaneously, they form this cluster of pupa cases at the top of the cereal heads or the forage grass heads. And a lot of people were seeing these in mid to late July last year and wondering, what are these eggs all over the top of my the heads of my crop? And the answer was they weren't eggs, they're pupa cases of a parasitic wasp. And so I think we had some decent levels that did get into the population eventually. So grasshoppers, this is one that I'm going to suggest you need to be watching for this year. And this might be one that down the road we need to pay more attention to as we get into possibly some drier cycles. We do a survey every year where we try to predict and forecast what will happen next year. And so what we do is we monitor roadside ditches in August. Last year populations were quite high, to the point where people were doing some spraying, either field edge or whole fields. When we did our survey in August, we produced a map, and this is what it looks like. And I always caution people. People like to call this the grasshopper forecast map. Really what it is, it's a map, it's a snapshot of what we saw in our August survey, but I don't like to call it a forecast alone. There's more information you need to add to make this a forecast. One of them being, what were the egg laying conditions like? What were the overwintering conditions like? And what were natural enemy levels like? And we had very good egg laying conditions in August. We had very little rainy days, cool days, so they had good egg laying conditions. I don't expect much winter kill. We did have a few cold snaps over the winter, and there wasn't a lot of snow, but the pet species lay their eggs a couple inches into the soil, and you very rarely get temperatures that deep that would kill the eggs. You need to get about -15 degrees, about 5 centimeters down into the soil for you to start getting significant grasshopper egg mortality. And you really don't want that, because if you do get those conditions, you probably are going to lose your winter cereals and things like that. So most years I suggest that we will likely have poor winter kill. The other thing too, grasshoppers tend to lay their eggs in areas like roadside ditches and areas that have lush green vegetation late in the season. And these are areas that are good at trapping snow. So we really don't suggest that in most years you're likely to get high grasshopper egg mortality. And a lot will depend on conditions in the spring, what populations do this year. I always like to put our map into perspective by comparing it to previous maps as well. What are the population trends doing? That can be important. And so what I'm presenting here are 6 grasshopper forecast maps from the past 6 forecasts. And you can see that starting in 2016, green on the map means lower risk. When you start getting yellows and oranges, and those are your more moderate risk situations. Red means really high risk. Luckily, we don't have much of that. But you can see that the early maps that I'm showing were mainly green, and then things started to pick up. And that's because starting in about 2017, we started to get some drier summer weather. And it took a year or two for things to really start building. But by the time we did our 2019 forecast, which is done in August 2018, we were noticing some significantly higher grasshopper levels. And my gut feeling is we're still in that trend where the population is building. I don't know that we've really started to decline. We are starting to see some natural enemy levels pick up. I saw more bee fly adults last year than I normally see. I saw more of certain species of blister beetles that target grasshopper eggs as juveniles. So they're building a little bit. You've got some parasitoid and predator populations climbing, but they've had good weather conditions to help them out. So do be looking for grasshoppers. And really quickly, I'm just going to mention that if anybody is looking for a way to save a little bit of money, if you're doing maybe some of your forage grasses or pasture areas for grasshoppers, there is a technique that was developed in Wyoming, where what they did was they took a high residual product and they put it on as treated strips instead of spraying the whole area. And so this diagram here, they're treating 100 feet, leaving 100, etc., etc., etc. And they found that they were still getting about 80 to 95% control when they did this. The reason being that grasshoppers, they are very active, they move around a lot, so they were moving from the untreated to the treated areas, and they were still getting good control at roughly half the cost. So this might be something to consider, again, especially if you're doing a lower value crop like a pasture. Here's some data. When they did it with Carborough, which maybe is a moderate residual product, they did it where they treated either the full pasture land at a fairly high rate or half the pasture at a half rate. They got 81% control versus 94. So that in many cases may still be acceptable. Malathion, their result was almost identical. Fipronil, a very high residual product. We don't have this here. We probably won't have this one here. But it is a very high residual product, probably comparable to our Coragen in residual activity. The difference in control when they did even a third of the pasture was really insignificant. So just something to consider. I know people who have tried this technique on cereal crops and soybeans as well, and anecdotally told me they thought they got good results. But as far as the research goes, I've only seen peer-reviewed research on this on pasture lands. So that's really where we have the data for this. But it might be something to consider again, especially for a lower value crop where you're needing to do some grasshopper control. And the University of Wyoming does have information on this if you're looking for more info. I mentioned some of the things that can take down grasshopper populations. So here's bee flies. They look like bumblebees almost. You might look at it and think that's kind of a weird looking bumblebee. That's a bee fly. We were seeing more of them. There's actually a species called the grasshopper bee fly that targets grasshopper eggs. That's all the larvae feed on. So I was seeing some more of them. Dark black or gray blister beetles. They will feed as larvae strictly on grasshopper eggs. I was seeing more of those last year. field crickets as well will feed on grasshopper eggs. They've been known to dig up grasshopper eggs and feed on them. So if you see a lot of crickets around an area that you know is a likely grasshopper egg bed, that could be what they're doing there, is digging up those grasshopper eggs and feeding on them. So we've luckily had some higher levels of some of the grasshopper predators and parasites. The thing that can really take a population down quickly is a pathogen called Entomophaga grilli. And you'll know you've got this if you start seeing dead grasshoppers clinging to the top of your plants. And we saw a little bit of this last year in late June, early July. It never really took off. This pathogen kills more grasshoppers when we get wet, damp summers. So again, we've had 4 dry summers in a row. If we get a wetter, damper June and July and August, that could help get this pathogen going. Even a few weeks of wet, damp weather when the grasshoppers are out could help get the pathogen going. But if it continues to be dry, which they're predicting could be the case, we have to prepare for this to be a potential long-term concern. So to summarize, cutworms and grasshoppers, they can successfully overwinter here. You need to be prepared for those and scouting for those. For cutworms, starting early in the season, as soon as the crop is up. Grasshoppers, start your scouting in June. That's probably when most of the egg hatch will be starting. Armyworms, seasonal migrants, will have pheromone traps out. But in most years, when they do arrive and become a problem, they're arriving in usually about early to mid-June and becoming a problem in early July. So start your field scouting with looking for larvae around mid to late June. That's a good time to be poking around, looking to see if the armyworms potentially are starting to increase. And again, grasshopper levels have been increasing. Can't stress that enough. Do scout those field edges early in the season. And we will end with that. And I don't know if we want to take questions now, Terry, or save that for the end.
We're going to save those to the end, John. But thanks very much for your presentation.
Okay, thank you.
Great. Now we'll get John Hurd on deck. And while John's presentation is being switched over to, I will read out John's questions. John, of course, is our provincial soil fertility specialist. And we got 2 questions for John. Given the high price of phosphate fertilizer, what are some things to consider? And what environmental conditions warrant the adding of potash to high potassium testing clay soils? And so those are John's 2 questions. And we'll proceed with that as soon as John is ready.
Okay, John, I see your screen. If you want to bring up your PowerPoint, not that, there you go. Yeah, PowerPoint. No, there you go. You're on mute, John, as well.
Okay. You can hear me now?
I can, and if you want to just, yeah, bring up that, I can hear you.
Okay, but you don't see my screen, do you?
Not yet, no. So, okay, so do you want to click on which presentation you want? Are you seeing it on a different screen, John?
Oh, I apologize for this.
Oh, it's all right.
Let me shut those beggars down, okay? And
go down to that PowerPoint presentation again.
yeah. Oh. I am clicking for all I can.
Okay, click that. Okay, is it on a different screen, John?
Yes, it is.
Can you drag it over to the other screen? All right, there you go. And then open it and then slideshow. If you want to just head up to the slideshow or there, that works too. Yeah, no, that works where you were down below. Once you can get to it. Okay, I can see your presentation. Can you hear me, John?
I can hear you. And
Okay, so I'd like you to go resume slideshow, what's showing right there. There you go. And hold on for a minute. We're just gonna wait. No, you need to drag it back over if you can. I've got one more option here. We'll get this right. All right. At the top slideshow where it says slideshow,
At the top.
right there, slideshow. Yeah, you're right beside it. Yeah. Click X. Close that out. Now go back up to slideshow. From beginning. And we'll just wait for a minute. Can you go down to your PowerPoint icon at the bottom toolbar and choose the right side? Yes, click that.
Perhaps slideshow.
Maybe you got to let someone else go. Obviously, this ain't working. I'm going to pull plugs and start over again, okay?
My suggestion, John, is just close your PowerPoint, reopen it, and we'll come back to you. So we can go to David Kaminsky then now, Terry?
Okay, then we'll load up David's presentation and I'll introduce David. So we'll proceed with that. So we'll come back to John. Right now, we'll go with David Kaminsky, our provincial pathologist in ARD. And for David, we have a selection of questions. One of them is, can you give an update on Verticillium Stripe, the differences between Verticillium Wilt and Stripe? And also, we want David to give us a summary of the new research on fungicides and soybeans, if it's profitable, and what are the resistant risks if we use fungicides on soybeans every year. So thanks, David, for being ready to jump in, and I'll let you begin now.
Okay, thanks, Terry. I hope I don't have the same concern as John. Can you see my title slide?
Yes, you're good to go.
Okay. Well, I'm going to talk about both soybean and canola diseases and a little bit of our survey updates. But since Dane Frey's left off with Verticillium Stripe, I think I'll jump straight to that. And in our survey, we had one column for Fusarium root rot and Verticillium Stripe. Hence that confusion about whether or not we were seeing it in the field. And we had a lot of summer students who were a little inexperienced perhaps in identifying this disease. But this is Verticillium Stripe. And I think Earl Bargan asked me, is Verticillium Stripe the same as what we were calling Verticillium Wilt a few years back, when it seemed to be a brand new thing here? And the answer is yes, it is the same disease. However, we call it Verticillium Stripe in Western Canada because we have not yet seen symptoms of wilt. That has been much more common in Europe, in the Scandinavian countries, where they grow winter canola. And so the disease has much more time to develop on the crop and cause wilt. Now, in this picture, you can see some rather diffuse discoloration within the stem that's cut near the base of the plant, where we'd normally be looking for blackleg incidents. But you can also see little black dots throughout the epidermis there along the side, and some shredding of the epidermis, which could be typical of another disease. Sometimes if Sclerotinia stem rot gets started but then stops, we will see a brown lesion with some shredding as well. And to look a little further at Verticillium Stripe, we'll look at a new infographic that Canola Council has just posted to their website. And first of all, why do we call it Verticillium Stripe? Well, the earliest stage or the earliest symptoms that we will see are this one-sided striping along stems, kind of brown on one side and green on the other. There are some abiotic conditions such as sun scald, which can mimic this, so you have to be a little cautious about just relying on that symptom. Those agronomists who have been around for quite some time will remember the early 2000s, 2002, 2003, when we saw an issue with a disease called Fusarium Wilt, and that also had this one-sided symptom. But that was dealt with through varietal resistance built into the plants, and we really haven't seen Fusarium Wilt for many, many years. This is now much more common to see this one-sided striping due to Verticillium. Again, here's the shredding symptom that we can see on the outside of the stem. And if we look at the outside, we should see those little black dots, which in other crops and diseases you might think are Pycnidia, a spore-bearing structure, but these are not spore-bearing structures. They're known as Microsclerosis or tiny sclerotia, and they're formed in many parts of the plant, within the epidermis, within the vascular tissue, and even within the pith, the central core of the canola stem. And here's a stylized life cycle. It could potentially be a wilt because Canidia, the spores of the fungus, do get caught up in the xylem tissue. Those are the vessels that take water and nutrients upward in the plant, so it can choke it off. Here's just a little bit closer look at the inside of the stem, showing those microsclerotia, both underneath the epidermis, within the vascular tissues, and within the pith cavity of the plant. Won't be a disease we'll be able to manage with fungicides, I don't expect, because of its growth habit. We're just not sure of the impact of this disease yet. We have seen it for a number of years. In this year's survey, I think we had indicated that from our survey of 160 fields, we had seen it in only about a dozen fields, but we're sending all of these stem samples to Shaway Go at PSI Lab, and they looked carefully at those stems, and they determined that somewhere between 30 and 40% of the plants that we sent, or stems that we sent, were in fact carrying this disease. It seems to, in our crops, develop later in the season, and it could be confused with a couple of other things which cause kind of a gray stem. One is actually called gray stem, and the other is the weakly virulent type of blackleg known as Leptosphaeria biglobosa. Here's the cross section of the stems. I'm glad Dane showed the variation in severity of blackleg. That's the one in the center. It's often these little wedge-shaped or V-shaped discolorations and quite dark, whereas Verticillium infection, what we see on the left, is quite diffuse and is right through the whole cross section of the stem. Here's a healthy looking stem on the right for comparison. Okay, now I'm going to jump to soybean disease survey, and this year we surveyed 66 fields across the province. This color scheme sort of represents the intensity of soybean production in different RMs, and so you can see where Terry is up in the Broken Head district. That is one region where we're seeing enough that we want to examine a couple of fields. Down here in the middle of the Red River Valley, we have Emerson Franklin, Rhineland, same kind of concentration, and a little further up in Morris and McDonald, lots of soybean production. So we would look at 3 fields in each of those RMs. 66 fields in total, representing somewhere around 9,000 acres, which is somewhat under, but we're shooting for somewhere around 1% of commercial production. This year, because of fewer surveyors working on this job, we decided to visit the fields only once at approximately the R5 growth stage, whereas before we were going twice, once at R4 and once at R6. We had about 9 surveyors from our organization and from the pulse growers who were involved. and Ag Canada is helping us by looking at the root rots. What do we look at in surveys? Well, there are 3 diseases we see year over year, and they are bacterial blight, Septoria brown spot, fungal disease, downy mildew, another fungal disease. We look for white mold, which is Sclerotinia. In this year and in this field, we saw absolutely none of that. And we look at some of the minor diseases which might be up and coming. In this field, the one that we saw a little bit of was frog eye leaf spot. We're also always keeping an eye open for these things, such as iron deficiency, chlorosis, and things like sudden death syndrome, soybean rust, which we haven't really seen over the years. Phytophthora root rot is one that we have been keeping an eye on closely. It has been an issue in wetter years, and as many have said, the last 3 or 4 years have been on the dry side. This is probably the R4 stage, so that was from 2019 when we're looking earlier. This is probably closer to the R5 to R6 stage, and this is a pathologist who works out of Dr. Dayef's lab. This is Ahmed Abdelmajid, and he has worked in the States and is quite conversant in all of the diseases of soybean, so it's been helpful to have him with us to point out some of the diseases that we don't normally see. This is me in a patch where we did see some Phytophthora root rot, and it is often in patches of the field where water has lain for some time. That's a little bit of frog eye leaf spot that Ahmed is showing me, and I think I have another picture with images in a moment. This is the 2 diseases that we see most commonly, brown spot and bacterial blight. The bacterial blight has yellow halos around the spots. Septoria brown spot is many more brown spots, and the only yellowing you get is in larger patches, sometimes the whole half a leaf, but around the edges of the leaf. It's not around each and every lesion. There are similar conditions which are conducive, wetter weather, of course, but these can be difficult to distinguish. One thing about distribution on the plant is that bacterial blight is often seen higher up in the canopy on the top leaves, whereas brown spot seems to begin in the lower part of the canopy and work its way up. This is that 3rd disease, which we see quite commonly. It's downy mildew. Its parent is yellow spots on the top of the leaves, which when you flip them over, you'll see this felty gray to brown growth, which is spore production on the underside of the leaf. And again, you can compare that to bacterial blight, which is much more common. What did we see in the 2020 disease survey? Well, you can look at the prevalence, that is the number of fields in which we found a disease, highest with bacterial blight, next with Septoria brown spot, and very little downy mildew. The next column is incidence, which is the percentage of plants in the field showing that symptom. And the final one is severity. And we rate all of these on a 0 to 5 scale, so you can see the levels of infection with each of those diseases is quite slight. I've highlighted the Eastern and Interlake region, where we surveyed about as many fields as we did in the Central region. And you can see for yourselves how that compares with provincial averages. Here are some of the minor diseases, and you can see that in your region, we saw no Sclerotinia whatsoever. I'm sure there were individual fields that we did not see that had some, especially since some of the area was wetter later in the season. So some of the later infected, or sorry, some of the later maturing crops would have seen a little bit of white mold. Frog eye leaf spot was seen everywhere at limited prevalence, and again, quite low incidence. I was asked about whether there's current research going on the use of fungicides in soybean, and yes, there has been. Just the other day, I was very fortunate to receive a report from Kristen McMillan, Some of you may have known her as Kristen Podolsky. She is now our research chair at the U of M, and she works in the Soybean and Pulse Agronomy Lab. She's been conducting a trial here at Carmen over 3 years with 2 different fungicides in both single and sequential applications, and she has found in none of those years that there's been a significant yield response. So the cost to the grower would have been the use of those fungicides with no result. But you have to note that we've had 3 consecutive dry years, and the amount of disease that's been observed in those plots has been quite limited. We also have these trials going on farms, grower cooperation with Manitoba pulse and soybean growers. And in 2020, there were 7 sites where they used 3 different fungicides, and again, no significant yield response. Septoria brown spot, that one disease I talked about, was the one found in most of those fields, but at quite low incidence, of course, less than 2 on that 0 to 5 scale. And this on-farm network has been doing this kind of work for about 6 years already, and over 66 trials or site years, they have only seen about 15% of those cases where there has been some significant yield increase, but that has only averaged 0.7 bushels per acre, which really, again, doesn't justify the cost of the fungicide use. I'll review the disease triangle, what leads to disease, a susceptible host, and soybean has not really got significant resistance to any of those 3 foliar diseases that I've talked about. You have to have the presence of the infectious agent, and with a tight rotation, you can expect that it's going to be there, or they are going to be there year over year. But the third thing is a favorable environment, and without that, a wetter year, that's going to be quite reduced, and the amount of disease is going to be slight. Was also asked about what is the risk of resistance, fungicide resistance in soybean crops with the repeated use of fungicides. And the one group that I'll single out as being at higher risk of the development of resistance are the Strobolurins, also known as group 11. And one example, I'm sorry that I've singled out a few fungicides, but these are the ones that control both Septoria brown spot and white mold or Sclerotinia if they're in the crop. Acapella is Strobilurin, a group 11 fungicide. And one thing I'll point out is that it's registered over a wide range of crops, including cereals and canola. So some growers might be using it in the intervening years between soybean crops, and that puts them at higher risk of resistance of fungicide, resistance to fungicides. Many of the other products have a mixed mode of action, that group 11 with either group 3, which has a low potential for resistance development, or group 7. And all of these with their mixed mode of action, we would have to say that we have a lower risk of fungicide risk developing, resistance risk developing. I think that's all the slides I have for you. This is my contact information. If you have questions based on what I've said or other plant disease concerns, I hope I haven't gone over this too quickly, but I want to leave some time for our other producers. To our moderators, were there any other questions that came in this morning?
No, we're going to move on to our next speaker, Dave. Thanks very much for your presentation. So we'll check in with John Hurd.
Okay.
All right, I'll take myself.
Okay, John, we're up and ready.
I am. I can talk, and am I showing?
Yeah.
Yes. Do you want to just head down to do a slideshow just a little bit further right, John? How you put it in? Yeah, you got it.
I got it.
Right,
very good.
We're just delayed. It's not showing quite yet.
Let me keep.
There we go. You're good to go. Carry on.
Okay.
Yeah, go ahead, John.
Yeah, first of all, that I apologize, Terry. I was queued up and ready to go, and on this topics that you put together for me, questions from your growers and others on phosphorus and potassium, and nothing else, because I don't want to use up any more time. But the something else that's been distracting me and why I missed my cue is this, that the provincial variance that was prohibiting fertilizer, nitrogen, phosphorus, and manure application has been lifted as of 1 minute after midnight, I guess. So it is legal. It doesn't necessarily mean that you have to race out and put your fertilizer on now, but it means that they've assessed the risk of runoff. The risk is very low because the soils are not frozen anymore. There's no snow around. And so if you are wishing to apply manure and you are wishing to apply fertilizer, you may now proceed. But again, stop if it looks like it's going to be running into the water. Okay, dispensed with that. The questions that came forward is dealing with the high cost of phosphorus, which is heading up, and what are options for growers? So we'll cover those. And then the other was, what about soybean response to potassium on high clay soils? I'll finish with that. So my comment on the phosphorus is, well, what's new about that? Veteran growers have seen this in the past. Fall 2008, phosphorus prices spiked up over $1,000 a ton here. I have the phosphorus on the graph on the right. If I can get the marker to cooperate with me. Right here, you could see where prices spiked over $1 per pound of P2O5. And how did farmers in Manitoba react to that? I also track the fertilizer phosphorus, what was removed in the crop and fertilizer applied within the province. This doesn't include manure, but it includes fertilizer. And we could see that, yes, there was a reduction in application rates the spring of 2009, following that run up in price. So farmers do respond by backing off rates somewhat, but you'll notice that the rates do not disappear to zero. It seems to be a rather subtle reaction to that. What has had more impact than that is probably during this period of time, we were having a lot more removals than application. And that's the soybean effect largely, that and high canola yields. So we saw two things going on. We saw that backed off fertilizer rates a bit, and we were growing some crops that we were not putting much fertilizer on. So our removals usurped our application rates. The other part of this was, I said, well, hopefully we fixed that. You can see that in the past couple years, application rates have actually been higher than removals. And I consider that a good thing. That's making up for the soil carried growers, or carried the province basically in phosphorus through those years of soybeans when we're still learning about life of soybeans. And so in the past, growers have made the opportunity to replenish some of those depleted soils. I hope that was part of the plan. Now, the question was, what about people dealing with these high fertilizer prices? You know, when I talk to dealers, over 80% of the fertilizer phosphorus was already purchased or applied in fall. So there will be some that are dealing with these high fertilizer prices, but certainly it's a minority. But to me, it's a good thing if people treat phosphorus with the respect it deserves, as if it was expensive. So it's not a bad thing that we treat phosphorus respectfully as if it is expensive. So my strategies are, and I'll run through some of these separately, is the soil test. Meter out your phosphorus rate by the soil test and taking into account your past management, whether you've been replenishing those fields, building, maintaining, depends on the crop you're growing and also the crop sequence. I'm going to direct you towards our sufficiency rate approach, talk about the importance of seed or close to seed placement, and then some less preferable options. You know, just wait till the soil warms up and you don't need much phosphorus. But then we also know that that is going to curtail most of our yields. And then I've got just a very minor amount to talk about the others. So first of all, we use this classic graph to explain a lot of our phosphorus approaches right now. One is our replenishment or long-term approach, but today I'm going to use it to explain the short-term approach for you, where these are the average 5-year or 6-year yields, because phosphorus response is rarely a curve like this, but averaged over 6 years it was. And we can see that in this study, 3 soil test levels were essentially produced. One where there was no phosphorus applied early, some where there was a moderate, and some where there was a high rate applied early. And so on those soils where phosphorus levels have been built up, there was modest response to seed-placed phosphorus. You know, 10 pounds, a low rate might give you the starter effect you're looking at, but there's not a big response to more. But if you're in soils that have been depleted, lower testing range, not only do you need these lower starter rates, but for full yield potential, you need to maintain those rates. So you talk about wiggle room with phosphorus rates. Your wiggle room is on your medium high test soils. Your wiggle room is not on your low testing soils. And where are those low testing soils? If you look at some of the AgVise data that they've had in the past, looks like in the eastern Interlake area, 1/3 of your soils in that area are testing low. So on those soils, I'm saying there's no wiggle room. You've pretty well got to keep your rates healthy or at normal rates, and your wiggle room is on your higher testing soils in this, where you could reduce rates somewhat without impacting yields. Because we know, here's an example of a canola response to phosphorus. So canola is a crop that responds. Wheat is a crop that responds. This is just to this all has phosphorus fall banded, but this is the response to a low rate of seed placed in the spring. We have corn that responds to phosphorus. And so those are examples of crops that respond. Non-responders are crops like soybeans and flax. For those, we can wait a while to replenish the soil reserves. Something else is the rotational effect. I'd suggest that if your crops are following canola or fallow, particularly if it's corn or something like flax, there's no wiggle room there. If we're growing corn on canola stubble and we have lower testing soils, we need that phosphorus. This is from the studies done in University of Manitoba. Here, corn after soybeans, and we can see that the check plot, it's still growing well, but it's behind in maturity. But here, corn after canola, it looks like it's about a month behind. So that's where you really need to use your full rates of phosphorus fertilizer. The two approaches, we now have two approaches: the historic, our short-term sufficiency approach, our long-term sustainability approach. This is what we like to move people into. But in years with high phosphorus fertilizer costs, then we tend to revert to the short-term sufficiency approach. That means that we are applying enough to get good yields, but we're essentially drawing down soil reserves. And that means using our table out of the Soil Fertility Guide, which is some pretty modest rates and rates of fertilizer that often are below the removal amounts. The type of opener you use makes a difference too on how effective seed-placed phosphorus is going to be. Tight placement with disc drills or narrow openers is going to be more effective than wide row spacings. And for this, we go back to where they use some radioactive in some Manitoba studies, some radioactive phosphorus to find the uptake. And with a press drill Where there was tight placement of phosphorus and seed, we got really good uptake of early season phosphorus because it's so close to the seed. Where it was broadcast or disker phosphorus, and with the disker, that would be similar to what a wide hoe drill would do. The phosphorus, it's less intimately placed with the seed, and so it's just less effective. So again, in this case, drills with low seed bed utilization tend to maximize the effectiveness of low rates of phosphorus. So we've covered these strategies. Again, I don't think that's one that may impact yield too much. I'm not going to slag the enhanced efficiency fertilizer microbials here today, just to say that refer to some of Cindy Grant review and presentations. Much of the work done by Dr. Cindy Grant showed, and you could go to this presentation at the MAC 2010, there's no yield benefit observed from using Avail or Coated MAP, Jumpstart, or microbial inoculants. So if you choose to use microbials, just be aware that it's not making phosphorus in the soil, it's simply drawing down your reserves, and you'll need to make those up. And I finished my phosphorus with this slide saying that if you're going to draw down reserves in the short term, a short-term survival approach, you also have to be watching the price ratio, and that is the price of your crop versus the price of phosphorus, to know when is phosphorus a good buy, when can I buy lots of phosphorus per bushel. And I'm sorry that train kind of left the station this fall, but they've been good opportunities the last couple of years, and it's important to be able to recognize these opportunities in the future, when to be in a replenishment phase, so that you can also take advantage of a drawdown phase. That's all I got on that. Potassium uptake. Before I talk about potassium on the clay soils, first we need to recognize that potassium uptake is due to two factors I'll talk about here. One is soil reserves, which tend to be high on our clay soils, but the other is the mechanism of movement, which is diffusion. Potassium diffuses like phosphorus to the root over short distances through the soil solution. When soils are dry, that soil solution is not continuous, and diffusion rates can be slow as potassium moves through the water film. And that's why we tend to see responses to potassium on high K soils in dry springs, predominantly not on soybeans, but on cereals. And if we look here in your area, you only have less than 20% of soils are testing low in potassium. I'd expect that they're your lighter textured soils. The clays generally have high reserves, but again, they are affected by the environmental conditions, not just the amount of exchangeable potassium. If we have deficiencies in soybeans, they show up really textbook with this yellowing on the leaf margins. So that's early season deficiency. It can also, we've observed very greatly within the field. Rick Storchuk from the East there provided these pictures, I think, on some lighter soils, and we can see how there could be patterns. Severe deficiency can, and based on some type of past management or soil differences. Late season, we can see these deficiencies on the leaves, and what it is plants pull potassium out of the leaves and move to the seeds for seed filling. Because soybeans tend to remove quite a bit of potassium per bushel. In this showing, just a pound and a third of potassium per bushel. So of the crops we're growing, it's probably one of the higher removal amounts. So we had a lot of studies done in Manitoba and had very unspectacular response to potassium, even on low K soils. It seems that soybeans are pretty good at acquiring even limited soil K amounts. But if you are choosing to apply, avoid seed placement. The salt effect is hazardous to the soybean. And a mounting body of evidence coming out of Minnesota, Indiana, other places, that very high application rates of potash appear to be reducing some soybean yields, perhaps due to the chloride level, unsure, but this is a bit of a heads up that probably not a good thing to bulk spread or build soils directly before soybeans. So if you're looking to apply rates to, for example, match removals, consider doing elsewhere in the rotation. Corn, forages, and cereals are probably all more likely to respond to potassium than what we've observed with soybeans. And that's all I have to say about that, Terry.
Great, John, greatly appreciated. And I'll just remind the audience, please enter any questions in the questions tab, and we will be addressing questions at the end of all of our presentations. So now I'll ask Kim to get her presentation on deck, and we'll move on to our next one. And that's Kim Brown Livingston, our provincial weed specialist. And the question we had for Kim was, how can producers respond to glyphosate resistant water hemp with a particular focus on corn and soy?
I'm just going to interrupt you for a quick moment. Kim, can you go up to the word display settings at the top there? And click that, please. And click that. And then swap.
Oh, sorry. I had them mixed. Sorry about that.
There you go.
Thank
Good
you.
to go.
Thank you, Lori. Okay, and thanks Terry and Ingrid and Earl for getting this going, and thanks everybody for listening to me today. Just gonna get going here. So yeah, I just want to talk briefly about water hemp. Oops, sorry, my slides aren't advancing. I don't know why.
Click your screen. There you go.
Yeah, there we go. Okay, so tall water hemp, and we've been talking about it a lot. My predecessor Tammy has done a phenomenal job of getting this going, and unfortunately it's been found in the East Interlake area, and there's been more found in Manitoba. I'm going to tell you about that in a little bit. But basically it's in the pigweed family, and anytime you see a pigweed, go take a look at it. If you think you have a patch of pigweeds, or you think there's something in any crop that got missed, or along a fence line or along a ditch, please investigate and figure out which pigweed it is. We need to be on top of this because this thing is pretty scary. So it's an annual crop, a C4, which means it's a warm season crop. So it's basically got the same life cycle type thing as our corn and soybeans that I'm going to be talking about today. It's dioecious, which means male and female plants. I'll get to that in a minute. It's genetically diverse because of that. There's a lot of basically outcrossing and stuff because of that, because of it's a dioecious plant. It produces on average about 250,000 seeds per plant. A single plant left undisturbed could produce up to 1 million, but in seeds, which is a lot, but in regular crops, usually we're looking at that 250 to sometimes 400,000 seeds is what you see. Germinates all summer long, especially in reduced tillage systems. And I have seen pictures of tiny little ones that are only 4 or 5 inches tall that still have a number of seeds in them. Whether or not those will be viable, those seeds are still there. It can get up to 8 feet tall. And a big thing on this one is that it has a smooth stem and kind of shiny, glossy leaves. Like I said, when you see a pigweed, go look at it. Red root pigweed, if you look at the bottom picture, is fuzzy, and water hemp is not. It's very, very smooth, and it does tend to be a little bit darker and shinier leaf, but not necessarily always the case. But the hairs are a dead giveaway. There's very good YouTube videos. My predecessor, Tammy Jones, has a really great one. There's lots of videos on identifying red root pigweed, tall water hemp, Palmer amaranth, that type of thing. So please familiarize yourself with that and know what we're looking at. I do have more pictures, but I haven't got them in the presentation just in because of time's sake. This picture here, how many amaranth seeds are on this dime? And the answer is about 70. That makes finding contamination in crop seed lots really a challenge. And If you look at the picture on the right, there's a little Energizer AA battery there. There's a little vial of seeds and how many seeds are in that container. And that's just about almost 44,000 seeds in that container. And you can see seed contamination is a major concern. And so again, this stuff, once the seed gets around, and it's really, really hard to get out of seed lots. This is a map of North Dakota, and this is from Joe Eichley, who's at NDSU. He's a professor at NDSU, and he is basically showing you where it has been found in North Dakota, which is pretty much everywhere that touches our border. So we have really seen this more concentrated more in eastern Manitoba, but it definitely has the potential to be all through Manitoba based on where it's coming from. This is our water hemp distribution map in Manitoba. This is the 2019 map that are confirmed in municipalities, and we can see definitely that East Interlake area. There's been some confirmed in Dufferin, some down in the arm of Rhineland, which is down south there, right on the border. And I myself found one in Dauphin, which is the circle there up in the north. Just got called out to a field. A farmer was questioning why there was a 7-foot tall plant in his field, and he thought he should call someone. And I said, yeah, absolutely. If you find a 7-foot tall weed, please call someone. And I went out and I investigated, and it was one single plant. We are definitely checking the field and the area next year. And I feel it came in contamination in the seed lot. That's what my thinking is, but we're not sure on that. But either way, Dauphin has been confirmed, and we will be updating the map to show that. So yeah. And that's us there and here in Carmen. Basically, it's surrounding us, and I fully anticipate to be finding more confirmed sightings next year, or this coming year. This is off the an international database. This is the resistant speeds, and this is the top 15 weeds in the world that are basically resistant because of the number of sites of action that they're resistant to. So these are the weeds that are resistant to more than one mode of action. And the 3 that I've circled there, one of them is wild oats, which of course we're always dealing with here in Manitoba. But the very top one is Palmer amaranth. That's resistant to 8 different herbicide groups. And the bottom circle there, this one here, is our tall water hemp, and it's resistant to 7 different herbicide groups. And there are populations that are resistant to multiple groups within that. As far as I know, there isn't anything that's resistant to all 7, but it's close. There are populations in the United States that are resistant to 5 different chemical groups. It's getting pretty thin on what you can spray at that point. This is again from the same weed. This is the International Herbicide Resistant Weed Database. This is a little closer. This is in Canada now. And this is the cases in Ontario when water hemp was first found and what it's resistant to. And I know that this is Ontario. This isn't Manitoba yet, but I'd like to show you kind of what's coming at us. And starting way back in 2002 and going up to 2017 is when some of these populations were found, either in soybean or corn is where the water hemp's been found in. And it's been found resistant to atrazine and to imazethapyr, glyphosate and imazethapyr, atrazine, glyphosate, imazethapyr, and lactofen. That's an older group 14 chemistry. We're not using that here in Manitoba, but we definitely do use some group 14s. This population here was resistant to atrazine, glyphosate, and imazethapyr, and this last population was resistant to atrazine, glyphosate, and metribuzin. So any of you people growing corn and soybeans, those should be triggering. Those are chemistries that we are using in our corn and soybeans, and we use them a lot. And again, so we see anywhere from resistant to 2 to 4 sites of action, modes of action, and it's, you know, your group 2, your group 5, your group 9, that type of thing. Those are the ones we use a lot. So here, this is a picture of tall water hemp. This is it over here. Again, looks very much like a red root pigweed, but it's basically red root pigweed on steroids. The growth rate on this is 50 to 70% greater than many other annual weeds, and there's multiple herbicide-resistant biotypes, and the resistant groups to groups 2. So that's anything from your sulfonylureas to your IMIs, which is what we use in a lot of our pulse crops. Group 4s, that's your 2,4-Ds, your dicambas, that group, not necessarily necessarily those particular chemistries in that group, but that's that group of chemistry. Group 5 is your atrazine type thing, your atrazine, your simazine. Group 9, that's your glyphosate. Group 14, those are PPO inhibitors. Those, we are starting to use a lot of those, and I'll be talking about that in a minute. Our group 15, these tend to be some older chemistries. Use some of them, have used them in the past quite a bit in corn. And our group 27, which is our HPPDs, those are our bleaching chemistries. It's been identified at 5 locations in Manitoba, and what we need to watch for is basically rapid plant establishment along field perimeters, standing water, drainage ditches. Just because we're heading into a fairly dry year, that doesn't necessarily mean we're not going to have water hemp. By the name, water hemp does tend to like, we do tend to see it in the areas where they're more moist environments, but it will grow in areas where it's dry as well. And there's another weed, Palmer amaranth, that tends to like the drier sites a bit better, but you will get both of these. And luckily we don't have that yet, or much of it. Maybe the occasional plant here and there that's been found, but we are watching for both of these weeds. They're both in the amaranth family, and just because the name says water hemp, that doesn't mean it can't be found in the drier areas as well. There was some herbicide resistance testing done on some of the populations that were found in Manitoba in 2019. There were 6 different fields tested, and within that field, 5 different plants within each plant within that field. In red, this is group 9. That's our glyphosate. Basically, almost every single plant that was tested was resistant to glyphosate. So that's not great. In yellow, not quite as bad, but still pretty bad. This is our group 2 testing, and this down here, that's just the particular type of test or the particular type of resistance. But in this field, 2 out of 5 samples, this is 4 out of 5, 5 out of 5, 5 out of 5. So it's pretty bad. So basically group 5, which is our atrazine, a few other chemistries in there. So far, nothing in Manitoba. There was a question mark with this one, but it still did not come up as definitive resistant to group 5. Our group 7 chemistries and our group 14 chemistries so far are still working very well, which is good. Again, this is a very small data set, but this is just what we found, and this is Manitoba numbers. So one thing too to note about water hemp is it's dioecious, and there's going to be male and female plants. And the female plants tend to have these long, spiky things with all the seeds on them. The problem with this is herbicide resistant comes on a lot faster with this type of a plant, because a female plant that could be susceptible to the chemistry that we're trying to spray, like to glyphosate, it might be susceptible to glyphosate or group 2s. But if a male plant is resistant, and if some of that pollen fertilizes the female, then the seeds that female produces, some of them will be resistant and some of them will be susceptible. And so that is how a resistant population grows a lot faster in these type of plants. And that's why it's really becoming a huge problem south of the border. So with herbicide layering, basically we need to be using multiple active ingredients from different herbicide groups to control the same weed in the same field in the same year. So not necessarily the same tank load. This is different than tank mixing. We still want to be doing tank mixing, but basically we want to be extending the level of the control throughout the season. We want as much control for as long as possible. It can provide improved weed control and return on investment in the absence of resistance. And layering could be used to reduce multiple resistance in one weed or address several weeds that are each prone to resistant in different herbicide groups. But basically, we're going to have to get a lot smarter with our chemistry, and we're going to have to use a lot of different chemistries and more groups. This is a slide from Bob Hartzler at Iowa State University, and in a traditional soybean program, not much is happening out here early in March, April, and May, June, July, all the way through to September, October. Basically, we've got a very long season crop going on here, either our soy or our corn. And normally, we're planting in May, and the problem is we tend to, with corn and soybeans, the weed-free period, the critical weed-free period on those crops is quite long. So they don't canopy in for a very long time, and we need to, our weeds can get away on us whether they're resistant or not. But basically, what we need to do is we can utilize some tillage here, especially with something like Waterhemp. It does better in reduced tillage systems, so a little bit of tillage here won't hurt. We apply a residual herbicide back in here or early on. This is where we use our pre-seed or our pre-emerge, and then we used our post herbicides as well. And hopefully we end up having a longer season of weed control within that field, within that crop. So what to do when you have water hemp? Our initial testing results indicate that you have a very good chance of it being group 2 and group 9. And I think we should kind of assume that going forward, that that's what we're dealing with. Even saying that, it doesn't appear to be a problem in competitive crops like wheat and canola. They just seem to be able to outcompete it and do really well. It's a serious problem in corn and soybeans, and this is where we need to look at herbicide layering to get the weeds out early when the crop is non-competitive, because your in-crop options become extremely limited. Our pre-herbicides, this is pulled out of the table at the front of the guide, our guide to crop protection, and I've pulled out basically these are pre-herbicides, and these are the ones that have residual activity. So these ones have a longer activity. These aren't necessarily these aren't your burn off products. Those are in a different table. Those ones normally don't have any residual activity whatsoever. So these products are actually in the soy table, in the actual table itself. So there is still burn off products, but these are the pre-products that we want to use that have some residual, and they're going to give us some extended control after we spray them. So we've got Authority Supreme, Dual 2 Magnum, that's an older chemistry. Edge is something that we don't see a lot of. It hasn't been used a lot down south. They are starting to use it more. And again, it doesn't have water hemp on the label in Canada, but it does on the label in the United States. And we would assume it would be very good. It's very good on pigweeds. Our Fierce and our Focus are a couple of newer ones for us. Heat Complete, Sencor and Treflan. Again, maybe we're circling around back to some of these older chemistries to use again. Our Treflan, our Volterra is a newer chemistry for us, and the same as Zidua, which is actually only suppression. That's our but that still has some activity. And if you look at, these are the active ingredients, and they're all big words, but it's kind of, there's not very many different big words in here. There's a lot of combinations, you know, pyroxasulfone, which is Zidua, it is in a tank mix, it's in Authority Supreme, it's in Fierce, it's in Heat Complete. You know, these, and when we look at our herbicide groups, we're mostly dealing with the 14s and the group 15s, and here's an older group 3, your Treflan's an older group 3, your Sencor is a 5 and a 3, but we are really dealing with our 14s and 15s, and that's pretty much all that we have that's going to work. When we get to in-crop, basically in the Red River Valley, we've got Reflex, which is a group 14 to be used in crop. We also use, there is a Hurricane and Ultra Blazer. They contain something called aceflurofin. I'm not super familiar with this, but I know it has been known to cause injury, so I would be maybe a little careful with that one. The Bazagran, which is Bentazon, that's a group 6 product that's in Viper. It's weak on pigweed, and that's probably being nice to it. It's probably very, very poor on pigweed, so it really isn't going to help you. So if you have water hemp coming up, there's really very little in crop to use that's going to work if you're outside the Red River Valley and you're not able to use Reflex, because your group 9, your glyphosate product's not going to work, and your group 2 products aren't going to work either. So this is where your Extend beans are going to work. That's our double stack trait on soybeans. That's our dicamba and our glyphosate. Again, so you'd have to be spraying, you know, can grow the Extend beans, and then you'd be using the chemistry. I would still fully expect you to be using a pre-product from the first table I showed you from a different group. And basically the products would be a premix here with Roundup Extend, or Ingenia, Extendamax, and Fexapan, which you would mix with glyphosate. But again, I would be using this system. This is my in-crop spray, and I would still be counting on a pre-spray with residual to get that layering effect. There's application requirements. We have to be very careful with this. We have to be good neighbors, and we need to be spraying this thing properly. There's also the Enlist E3 trait. This is fairly new, not a lot of varieties out, but they are coming, and I think in the future we're definitely going to see an awful lot of these. So these are soybeans that are tolerant to 2,4-D choline, which is a slightly different type of 2,4-D than we've had in the past. Right now we're dealing with the amines and the esters. glufosinate, which is your Liberty, and glyphosate, of course. And basically, Enlist Duo is the chemistry that you'd be looking at, and it's a premix of glyphosate and 2,4-D, and you can use that post-emergent, and you can also spray with glyphosate separately on its own, the 200 SN formulation. So when that becomes available, when we have more varieties for that, I think that's going to be going to be one of our only options as well. And again, you have to be very careful with spraying this, be good neighbors, and make sure we're spraying this thing properly. When we're looking in corn, here's our pre-products. Again, these are products that have the longer residual that we're counting on for weed control into that until that corn becomes a little bit more competitive. And takes out these weeds that just keep emerging. So our Dual 2 Magnum, again, that's older corn chemistry. Our Fierce and our Focus, which again, that's got some pyroxasulfone in there and a couple of different group 14s. Our Heat Complete, which is basically heat plus the Zidua, the pyroxasulfone component. Premextra 2 Magnum is a premix of metallochlor and atrazine together. Valterra, which is this flumioxazine, that's a group 14, and Zidua, which is pyroxasulfone, that's a group 15. So we're, again, weighted super heavy, group 14s, group 15s, that's kind of all you have. And another thing just to note, it's a different talk, but this pyroxasulfone, the Zidua, it's one of the few products we have left when we have herbicide-resistant wild oats, when we have group 1 and group 2 resistant wild oats, and sometimes we start getting resistance to group 8 as well. That's starting to happen. This is one of our last products we can use there. So we need to be careful. We need to look at how much we're using these chemistries, not just in our corn and soybeans, because we're basically using exactly the same group of chemistry in our corn and our soybeans. But we're also using it in other groups, in other crops as well. So we need to watch that. So in crop, your options are pretty limited, but Shieldex plus atrazine is working really, really well. That's topirolate, that's a group 27, so that's a bleaching chemistry, and also Armazon, same group, it works just a little bit different, but it's very good. They're both very good. Those are both group 27s. That's kind of pretty much all you've got. Your Liberty 200 on its own, your atrazine on its own, there is some level of control, but really not very much, and it wouldn't be enough. And your group 4 and your group 6 products, be very careful when using these. I would be checking with my seed supplier before using them. They are registered, they are in our guide, but there is because these are growth regulator herbicides, you have to be very careful using them with corn and using them properly and using the right rates and getting them in there at the right time. They tend to do better a little earlier on in the season. They just don't do well at the higher rates, and later on as the corn's getting bigger, it can be not great. There's some issues with these. Again, they are registered, but I would be very careful before using them, and I would be again talking to whoever you're buying your seed from and their agronomist and making sure that that's something that you need to be doing. Enlist corn when it becomes available, I think that'll be another great option to have in this system. That's probably a couple of years away before we start to see it, but again, very similar, be like the Enlist soybeans. It's another herbicide tolerant system, and that'll give us some more tools to throw at this water hemp. So with that, that's all I had. I know we've got questions at the end, and I just wanted to remind you the back page of our guide always has our herbicide groups and our resistance. And I think this issue is not going away. We're seeing more and more herbicide resistant in different weeds all the time. And again, we have to watch. We have to be careful with this and kind of know what we're doing on all of our fields on the whole farm. And that's all I have.
Thanks very much, Kim.
Thanks.
Greatly appreciated. A lot of information that you packed in there, and thank you. And we'll proceed on to our next presenter. So now we're going to switch over to Marla Reichman, who's one of our land management specialists. Marla had two questions provided to her. One is intercropping production practice ready for prime time and conventional crop production? And as well, where are we at with other regenerative agriculture practices as they relate to conventional crop production? So take it away, Marla.
Thank you, Terry. Okay, hopefully you can see my screen okay.
Looks great.
Good.
All right, so again, Terry had mentioned two questions that were kind of brought forward to me, one being related to intercropping, the other one to regenerative ag. And so we are going to review a little bit of this, and we'll do what we can in order to answer some of the questions, or in a way, maybe not answer them in full, because in some cases we don't really have the full answers yet, but there is some research that is kind of ongoing or coming down the pike anyway. So first question, is intercropping a production practice ready for use in conventional crop production? And so we luckily have a little bit of work that has been done through WADO, looking at a bit of intercropping. You can also look further west to work that is often presented by Lana Shaw, looking at what they've done in Saskatchewan on some intercropping. And quite often what they're looking at is just trying to figure out, what crops work the best together, what are the issues, like the technical issues related to either seeding crops together, spraying crops, the general kind of agronomy, harvest of different crops that are grown together. And these are a lot of the questions that maybe aren't being shown in the data that I'm going to present to you, but there are people who have been doing a lot of work on this. The first thing that to mention when we start looking at intercropping is that we always look at something called the land equivalent ratio. And that essentially is saying if you have two fields, if you've got one field seeded straight to peas and another field seeded straight to canola, that kind of typical average yield maximum that you would get on either of those fields should be equivalent to 1. Being that, like, what is the general equivalent? If you took those two fields now and seeded them to a mix of peas and canola, like in this slide demo, the amount of yield that would come off of those two fields in comparison to if one field had been just peas versus one field being just canola, that ends up being what they are kind of comparing in terms of this land equivalent ratio. So in this case, when WADO has looked at pea canola intercropping, in general, the land equivalent ratio is above 1. And that means that they're yielding slightly higher when seeded together than if they had been seeded separately. But there's varying degrees of which crop is actually performing well in this scenario. Quite often what happens is that the peas is actually the one that's performing better being mixed with canola. And I have questions too regarding, for those people who have actually grown peas before, they're kind of a pain to pick up off the ground when they do fall over. There's some benefit to having a canola hold those peas up. So is some of this being less harvest loss that's actually the full benefit versus the actual just higher yield? And that can be one of those questions that I don't know from the plot work that's been done on this, whether or not we've actually been able to answer that particular question. Indian Head has also done some work looking at the kind of general benefits, again, within a pea canola intercrop. Again, the land equivalent ratio of growing canola alone versus peas. And this is bringing again to that land equivalent ratio of 1 with varying nitrogen applications. And then they've done these mixes again with varying nitrogen applications, because a lot of the idea is that we can reduce some of the nitrogen that's being put down for the purposes of the canola, because the peas are essentially going to be grown without that nitrogen. So what can we end up doing? And in the wet years that they saw during this period, 230% of kind of a normal rainfall, the mixed row scenario where they had peas and canola growing in the same row generally outperformed. Again, it's the peas that are doing so much better in that scenario versus the canola itself. Quick mention of just some observation, because this isn't like a full data looking at across all of the treatments within the different reps and actually doing statistical comparison on it. One of the things that Scott Chalmers was finding when they were looking at the pea canola intercrop is that aphids were reduced where the peas and canola were grown together versus where the peas were grown alone. Whether or not this comes into an economic yield and hitting an economic threshold, that was not kind of carried out. And then the other thing that I'll show is work again with Kristen McMillan had shared this. Dave Kaminsky had also mentioned this annual report, and we can put the link to this annual report in the chat so you guys can have access to it. But this is looking at different legumes or pulse crops being grown in conjunction with other non-leguminous plants. So they were looking at peas, canola, flax and soy, peas and flax, and peas and oats. And in general, they find that the when you use the peas and canola would generally out yield everything else. The canola didn't grow very well in these comparisons as well, but they did find that even the canola on its own wasn't doing as well, I believe, because there was a lot of flea beetle and grasshopper challenges. This was from Arborg that they were doing this work in 2019 and 2020. This is just the 2019 data, but the 2020 data, but the 2019 data was very similar. So peas and canola were really the only intercrop that was consistently over yielding compared to the other crops. But when they looked at the actual revenue, peas and canola were not the kind of greatest revenue, and a lot of that had to do with the loss of revenue or the lower revenue with regards to the canola. A lot, again, because of the additional cost of growing canola and the additional pesticide applications required. But the pea oat and pea flax were the highest revenue here. And then followed by generally the pea canola. So obviously peas in terms of general revenue were doing a lot better than intercropping with something like soybeans and flax. I do want to make a comment though. Another interesting observation that they found when looking at this data was that they found that the pea flax combination here, there seemed to be a decrease in the prevalence of sclerosis in flax. And this actually makes sense when you think about increasing seeding rates or having something growing along with that sclerosis intolerant, let's say, plant like flax, because there's more other roots and crop using water in that seed row. And so it could actually be, again, they're doing this with full rate of peas plus a portion of the rate of flax. 3/4, half to a quarter rate of the seeding rate of flax. And so the flax is going to be doing better because, again, the peas are likely helping out using up some of that extra water, even though it was actually kind of a dry year. So that's what I have on just a little bit of information on intercropping. Again, there's more work being done on this. And in a way, we do need to, in order to fully understand how to, like how the economics is playing out, we do need to see a little bit more data, I would say. The second question was regarding where we're at with regenerative practices related to conventional crop production. And here I would say that we have even more questions in some cases. And my first question when I think about something like regenerative agriculture is the fact that we have various definitions of what regenerative ag means. Whereas when we talk about soil health, there are some very specific soil health definitions that are accepted and used and documented widely in peer-reviewed research. Whereas when it comes to regenerative ag, when I start looking up a lot of the different definitions, it varies, and it varies a lot based on maybe the interest of the group who is promoting regenerative ag, the kind of type of regenerative ag practices that are being carried out. But when you do look at these definitions or the principles of regenerative agriculture, most of them follow the 5 basic principles of soil health. There may be some additional things like looking at socioeconomic impacts within the system, that type of thing. But really what it comes down to is maximizing soil cover, minimizing soil disturbance, maximizing plant diversity, keeping a living root plant in the soil at as much of the period of a growing season as possible, and integrating livestock. And so in terms of what this means when we think about conventional crop production, we may not think about that idea of the integration of livestock other than, sometimes people might actually be grazing a cover crop, even though they may not have cattle, someone might want to bring cattle in, but there's costs with regards to setting up perimeter fencing to be able to do that kind of thing. integration of livestock can be the simple process of adding manure, whether you are like actually spreading manure, so not having the livestock there, but still bringing some of that manure, the recycling of nutrients back to the field. And so that is something that is still considered, whether it's a soil health principle or regenerative ag principle. But I do want to quickly touch on some of the kind of more major issues that I think before we get, and a lot of times when we talk about soil health or regenerative agriculture, we jump into the kind of the tiny details, the minute details of how to make a soil healthier, how to focus on biology, how to focus on a lot of those things. But we do that without first stepping through the other major issues that need to be addressed either at the same time or sometimes before that. And that can be things like erosion, that can be things like compaction, things like salinity, those issues that are affecting crop growth before we even get down to that microbiological question. And I know John mentioned earlier about the question regarding microbiologicals, and that becomes a big thing that we talk about with Regen Ag too. He said that any microbiological that's going to be helping you obtain more, say, phosphorus from the soil rather than adding the phosphorus is more efficiently mining the soil. And so with Regen Ag practices, sometimes people talk about, you know, not having to use certain fertilizers. We can make nitrogen, we can't make phosphorus, we make it more available, but we're still more efficiently mining a limited resource in the soil. So that is something that does get talked about a lot with regards to Regen Ag. But taking that step back to focusing in on, you know, what are the bigger picture issues that we're kind of skipping over sometimes when we jump into these principles. Keeping the soil covered or protecting it from erosion, again, is like steps 1 and 2, providing armor, providing cover, and reducing tillage are the main kind of first points that we need to focus on as we go through these different principles. We see more and more of this wind erosion happening. I'm curious what this spring is going to look like. It is drier than what we would call normal, but we've had a lot of these dry springs recently in many areas in Manitoba, and that means that we potentially will see those issues of soil blowing in the spring again if there's not enough cover. The active tillage we know can obviously destroy soil structure, and depending on the type of tillage that we're using, there's different levels of pulverization of the soil surface. Discs are obviously a lot more destructive of soil structure than something like a field cultivator. So that's something to keep in mind. And the other thing that I wanted to kind of showcase or talk about, because this is something that we did at Crop Diagnostic School a few years ago, was looking at the effect of tillage erosion or tillage equipment on moving soil particles around. And the reason why I want to bring this up, again, it destroys soil structure, and that destruction of structure means that you have a higher risk of wind and water erosion. But tillage itself also moves soil. And this might not seem like an issue for anyone who is on flat land, because you don't think about eroding soil down a hill like we have here. You can't see it, but the top of the slope, the bottom of the photo, if you were standing down here, you'd actually be looking down the hill. And there's a 2 to 5% slope in this area on the Carmen Research Farm when we did this demo. What we did was along this blue line, we trenched in about 3 inches wide, 6 inches deep, and we placed corn seed down in this, and the grain, the kernels themselves act as a portion or piece of soil. So when we passed over with the tillage equipment, we dragged that corn kernels along, and the kernels are found in each of the direction of tillage as far down as these little yellow X's. This is a 30-meter, 45-foot slope from top to bottom on the screen. And so there's a large distance that this has moved. Again, do we actually care if we're talking about flat land? Maybe not, because we're not necessarily moving soil off the top of a hill down to the bottom, but we do care if we're filling in drainage ditches on a regular basis, because it costs something to us to have to clean them out again. The other thing we care about is things like movement of clubroot or certain weed seeds and patches. If you think about how big a patch can get if we're moving things around with tillage equipment, this is a fair distance that some of this equipment can move soil. And note that when we were going up the hill versus down the hill, the distance is about the same in terms of the movement on this kind of lesser sloped land. So flat land, you're still going to have this kind of movement happening. This is just an image from a couple of weeks later when the corn had actually started to germinate. I took the little X's off. I don't have green X's here to show where the germinating corn was because it moved all the way to the top and the bottom of the slope in both, in all different tillage types, regardless of distance. So I just wanted to mention that because, again, it's something that we need to think about as a first step when we're starting to talk about moving into these regenerative ag practices. We do have some new tillage options that may help to reduce tillage. That being said, shallow disc tillage, like the high-speed shallow discs, are very aggressive. They are very detrimental to soil structure and can leave the soil at a higher risk of erosion, even though they don't till as deep. Speed is as bad as depth of tillage in some way for that erosion control or the erosion ability of a piece of equipment. And then strip tillage is something that people are starting to play around with now, looking at this combination essentially of tilling just in the seed row versus the kind of zero tilled area between the seed row. And that can actually greatly reduce some of the impact. So the question that I always get is, ultimately, if we're looking at regenerative ag practices or soil health practices, do we know that they're actually working? We can follow something like this that says, yeah, my soil is healthy, my soil is not healthy, likely, based on very simple observations that we see in the field. Does it blow or flow away? Does water soak in? Does it drain? Does it crust over? Is your crop growing well? Is it recovering the nutrients you're applying? Are there areas where the plants are dying, growing poorly? We can look at something this simple. We can also look at more complicated soil health tests, but there is what we have listed here is an endorsement of 19 tier 1 measurements. These measurements are things that the Soil Health Institute has said, you know what, we know enough about what these measurements tell us. Either the consistency of the lab results are working for these lab tests, or they actually tell us something about what's happening in the soil. That is great. We do have issues here in Canada with 2 of them, carbon mineralization and nitrogen mineralization. And that is because a lot of these tests are looking at things further south. When we talk about things like mineralization, we're often looking at things like the soil urendis test. Obviously, a soil that has more microbial activity and more mineralization or breakdown of carbon should be healthier. This comparison, however, this top pair of underwear that's very broken down is from row cropped corn just behind where you can see John and Diljit and I standing here, compared to a perennial forage that is 2 or 3 meters away from where we had planted the row crop corn, just beyond that little corn plot. And so this test is not telling me that the row crop corn is healthier. It's telling me that there's higher rates of mineralization of carbon, breakdown of carbon, decomposition because of the aeration from tillage and the nitrogen, high rate of nitrogen that's been applied. So we need to keep these things in mind. Soil health testing is sometimes difficult to work with. It needs to be able to identify differences, be stable over time. And there is a list of additional measurements that we don't know a lot about yet, but further study is required on these. So the Soil Health Institute is working on these, but please note that if you are sending soil tests away to kind of figure out whether your regenerative ag practices or your soil health practices are actually working, a lot of the soil health tests that are available at labs are currently on this list that don't necessarily have enough data yet to support the use of them to know whether or not they're great measurements. So that's what I have to kind of talk about. If anybody has questions, I'll be staying on to be able to answer them later. So thank you. Take it away, Terry.
Thanks very much, Marla. Greatly appreciated. Now we're going to move on to our last speaker of the day, and we're going to change gears a bit as Darren loads his presentation. We're going to have Darren Bond speak to us last. Darren is a farm management specialist because we're in the business of making money, and farm management is an important part of that. So the question we had for Darren was, with crop profitability changing as a result of increased crop prices in the last few months, how are risks on the farm evolving? So please take it away, Darren.
Thanks, Terry. Appreciate the opportunity. And yeah, things have changed. If you decide to go into your basement and not come out until there was a vaccine, and then checked your grain bids and fertilizer quotes, you might be in for a bit of a shock here. So whenever we start talking about planning and knowing where those risks are, we always go back to the foundation. The foundation is the cost of production. The ability to track your costs, whether in dollars per acre, and as we're pushing more producers to dollars per bushel, to determine where you stand, where your break-even prices and yields are, and being able to identify some marketing opportunities and profit thresholds. It's very crucial to do that. And as we're in times of change, it seems that we always need to be on top of these things to make sure that we're not leaving anything behind and not missing anything. Costs of production are very functional too, to determine our outflows and inflows when it comes to expenses and our sales. And lastly, allows us to take that to step back, assess some profitability, some returns on investments on the farm to see what we're doing, if what we're doing is making sense, and what we need to change. This is our cost of production. The lower corner there is a QR code. If you take your phone camera, hover over, it'll take you right to our website. I got the same QR code on the following slide if you're digging for your cell phone. But if anybody needs any of the hard copies, we do print a number of them. My contact information's at the end of the presentation. I'd be glad to send you some because they're great resources. And on our website, this is where they're found under the production economics cost of production section. Here's our fall 2020 costs, and this is what is up on the website and what we have for numbers. And we're going to essentially take this, compare with what we see today, and determine what has changed. And if we look at the bottom there in that red circle, we look that there is some decent profitability to be had with our old numbers. Now, I'll draw your attention to the price, the target price of 11.25 for canola and 11.40 for soybeans, and seeing those target prices, there's still some decent profitability with that structure. Jumping ahead, and I believe that we try to make farm management a little bit more fun. If you could see me, I'm using air quotations with that, but to make it a little bit more interesting to see how these things break down. And in the top set of numbers is our break-even price per unit, so a break-even price, and we can see the split out between it's what our operating costs are. So essentially in the canola line, we're saying that for every bushel of canola that you have, $6.07 goes towards the operating costs. And we can see our land costs and machinery costs in there, along with the labor and living allowances. This show us that in these set of numbers in fall time, we were just under 10 bucks a bushel for canola for a break-even price. If we were to flip that around and look that into a break-even yield, we could see, again, just using canola as an example, we're at about 24 bushels an acre just to cover our operating costs, another roughly 6 bushels each for land and machinery, another 2 for owners, labor and living, to give us just under 39 bushels an acre for a break-even all-in cost yield that what we'd need to see on the farm. So let's fast forward ahead to today. So what has changed? The big changes are the jump in urea and phos, bit of an increase in fuel, but the fertilizer costs have really seen an increase. But at the same time, we've seen an oilseed price rally here, and if we look near the bottom in our target price, this is new crop pricing of $13.50 for both canola and soybeans in this example. Wheat was a little bit higher, just over 7 bucks. And if we look at the bottom circle, what has changed and what we've seen is that this oilseed rally has outpaced the increased cost of inputs for canola and soybeans, but the cereals have been a bit more of a wash. We haven't really seen the increases there. Now, of course, if you purchased your fertilizer in fall time before these price increases, the cereals would show up quite favorably as well. But if we're looking at what we see now for our cost structure, that's what we see, is that this is mainly an oilseed rally, and this is something that I'm going to come back to and remind everybody to use the common sense and being grounded in what this actually means on our farms. So if we look at our break-even prices, we can see that, again, using canola for an example, our break-even cost is up a little bit. It's up on canola and hard red spring wheat, which is not a surprise considering that those are two large consumers of fertilizer, especially on a per unit basis. And what we can see at the bottom is our break-even yield is that we see in canola and soybeans, our break-even yield go down, which is a good thing, meaning that we can have a little bit more profitability, but it's largely unchanged in the cereals. It's up a little bit, but again, that showing that when we consider this, we really need to focus in on the canola and soybeans for how much things have changed in the last little bit. So we're going to get to the use your brain section of the presentation. I like to refer to it as, and especially when we start making decisions and start trying to decipher what this means on a go-forward basis. This slide here is our average yields from 2012 to 2020. This is MASC, Manitoba Ag Services Corp data. And what we've seen, and look at that top line of hard red spring wheat, we've seen a pretty decent yield increase, and it's trending upwards. Same on canola. Beans have been a bit flatter, although in 2020 we saw a little bit of a jump in yields here. But what I think this is important, and my colleagues have touched on this previously, is that we need to understand what this means, and especially when we look at hard red spring wheat, for example, as these yields go up, we have to feed the beast, if you will, when it comes down to fertility. That when we're starting to look at average yields above 60 bushels an acre, and it's thanks to the production management techniques of producers, but seed genetics, everything, crop production, everything along the line, is that we have to be really cognizant of what we need to do to continue to drive those yields upwards. And there's no cheating, if you will, when it comes down to that, especially on the fertility front. Jumping into land values, and I bring this forward because the last time we've seen things like this happening was back in 2012 and 13, started in the last bit of 2011, and we saw in those years 25% annual land value increases. This data is from Farm Credit Canada's Farmland Values Report, and once things normalized, we got back into the longer term appreciation averages, which is around 5%, give or take. And the question that I'm starting to get a lot more of is, what's going to happen in the future? Are these prices a blip, or is this the new norm? And I'm not sure if anybody could really answer that, but just to give things in perspective, the 5-year average for pricing on canola is $10.50 a bushel. It's about $6.50 a bushel on hard red spring wheat, and about $10.30 a bushel on soybeans. So we're looking at prices on the oil seeds, new crop oil seeds, be $3 a bushel higher, and that much more higher on old crop. So I think we need to take this step back and say, is this the new norm, or is this just the high point in the cycle? Crop rotation has a huge impact on profitability. A lot of us are locked into a certain rotation. We do have some flex acres, but when we look at it, oilseeds are a win with this new cost structure and this new pricing structure. Cereals a bit of a neutral perspective, but we have to ask ourselves, is there an ability to push that and the problems that come with that, and being aware of that, because oilseeds are only a portion of our total crop rotation on the whole farm. And judging by how much my phone's been ringing on issues like rent, from both producers and landlords. My concern is that this appreciation and this increased profitability that we've had in oil seeds is almost an overemphasis, or there's a disproportionate amount of attention paid to that and the increased profitability on the farm, when we consider that canola, for example, is only a portion of that crop rotation. So if you're thinking that, well, we're at 1350 new crops, so I have that much more cushion and I can be that much more aggressive. Well, and depends on your situation, but canola might be only a 1 in 3 year rotation, and if you have 2 cereal years, you're not seeing that increase in profitability in those cereal years as what you're seeing in the canola years. So you have to be very aware that oil seeds are only a portion of the increased profitability to the whole farm when we look at it over a multi-year situation. Land value, will we see those land value levels of appreciation? Many argue that land is already past its productive value, that often we see producers requiring 2, 3, 4 bought and paid for acres and using the profits from those acres to subsidize purchased acres. Where will this take us? And land value and land rent seem to be an elevator that only go up, and then maybe it might pause at a floor for a little while, but it seems like it goes up so easy, but then it just doesn't plateau for very long, and it doesn't go down. So we have to be very aware that, knowing our own costs of production, where does this put us when it comes to rental and land purchase decisions on a go forward basis. So just wrapping things up here, what are some take home messages? While there's greater potential for profit, there is also risk, and especially as we start to see the costs go up, and if that's going to be the new norm, our amount of investment per acre is starting to get considerable. We start looking at things like our operating costs, but I encourage producers also look at what your land investment costs are and also what your machinery investment costs are. If we start to see this land going up even more, we can start to see a pretty significant bite out of our gross revenue that's required to service that. And whether it be issues where if it's you're in a high equity, so most of the land's bought and paid for, there's still an opportunity cost to carrying that land. And I know producers will often talk about, well, you can't afford a train wreck, and you can't lose that the operating costs and the seed, fert, chem that you invest in that. It's also the amount of cost with this land is that we can't lose a year's worth of production. So we have to be very smart about things. With market volatility, I joke, I laugh. It used to be 5 cents a bushel was a big day that would have the coffee shop buzzing. Now we're seeing dollar swings, and people are just shrugging their shoulders and saying, COVID, China, U.S. political situation. There's a bunch of different reasons for that, for whatever's happened. But there is volatility, and it has made marketing, I wouldn't say more important, it's always been very important, but it has made it a little bit harder on the nerves. And then lastly, dryness. And then again, my colleagues have touched on this too. And this is something that, we need to be aware of, and the risks that go with this, and all of these, how they fit together. Again, being mindful of that, only a portion of this crop that we're putting in, if we look at it over a longer term rotation, is seeing this increased profitability, where the cereals are a little bit more of a wash, especially if we're looking beyond this year, and if these fertilizer prices hold and the relationship holds with the grain prices, is that we have to make sure that we don't start to get ahead of ourselves and think the farm is more profitable than what it is, just because we have a couple bright lights on the farm, where the whole farm is not seeing that type of profitability when we look at it in terms of a per crop or rotational view. In profitability, it's always great to invest back into the farm to do something purposeful with that profit, and generally paying down debt or creating a war chest or making some strategic investments, whether it be land or equipment, that have a positive return back to the farm, are all good moves to make. And I've had producers call and say, well, what's the best thing to do? And I would say, well, what's your weakest point? And that would probably be the best thing. But if you don't know, do a little bit of everything. And I think that's a good strategy, because at the end of the day, we all have to remember that what we do in these good times determines how we fare in the poor times. And I'm fortunate I get to see a lot of farm finances, and I'm fortunate to work with a lot of producers. And that's something that has been a reoccurring thing to me over the past decade, is that those who invested wisely, who saved, who paid down debt, who made their farm stronger, were just that much more resilient in the poor times, and often were able to capitalize on expansion opportunities in those poor times. So if you're not expanding now, don't beat yourself up too much about that, because there's one thing that I think is pretty true, is that things go up and things go down, and we have good times and we have bad times. So take the long view and keep your head on your shoulders and do those costs of productions, know where you stand, have a plan, and just use these good times to your advantage and increase your farm financial health, and good things will happen. So Terry, with that wraps up my presentation. I think you're going to open it up to questions now.
You bet there, Darren, and thank you for your presentation, and thank you to all of our speakers today for your presentations. You did a great job, and I think there was an awful lot of good information that was shared this morning. Lori, you were going to handle questions, so I'll turn it over to you.
Yes, I do have one, and this question came up during Dane's presentation. So Dane, why is the relationship between pH levels and clubroot infections, and why does clubroot persist in lower pH compared to higher pH?
Good question. I don't have an exact answer as to why it prefers lower pH than higher pH. Why do you prefer plus 20 degree days rather than minus 40 degree days? It's just more environmentally friendly for that particular pathogen. That's just how it likes it. Why the lower pH encourages a faster spread? I am not the scientist who would be qualified to answer that. I'd have to look into that and get back to you. But a lower pH does amplify the spread. It can develop quicker because that's the pH that clubroot is favored in. Between pHs 5.5 to 6.5 seems to be a sweet spot. Once you start getting over about 7.4, 7.5, from what I've seen in the field, those patches do tend to be smaller and they don't tend to spread quite as aggressively. Might also have something to do with soil texture. Higher pH is typically more clay or calcareous based. Those finer soil particles make it harder for water to move and swim through that soil structure. And as water and soil particles move through those pores, the clubroot spores that are attached to them have a harder time moving through a smaller space. And that just simply reduces the infiltration and spread rate. That may be one way clubroot is less favored by those higher pH / more finely textured soils. So if we're looking at an organic soil, like a peat-based soil, very low pH, those certainly are known hotspots where clubroot can spread and can be fairly aggressive. And those patches can get quite large quite quickly just due to the nature of water moving around in the soil. But the pH will also help increase disease.
Excellent, thanks, Dane. John G, this one looks like it's for you. If I had cutworms in the field last year, does it make it more likely to have cutworms this year? Did we lose you, John? You are muted if you are trying to answer that. Okay, there we go.
Okay, thanks. Yeah, so it's a tricky question, actually. It's a good question. Cutworms, the patches do move around from year to year. There are moths flying around in August laying their eggs. And so if they're in a field this year that really isn't attractive for egg laying, they may move over to a neighboring or another field that has a source of nectar for them to feed on. and some cover for them to rest in during the day. So just to give you an example, we've seen cutworm patches in the past that were in the previous year's thistle patches in a field. Crops like hemp can be really good at drawing in cutworms late in the season because there's a lot of cover for them. There might be some pollen or nectar to feed on. So it does vary, and those patches do move around from year to year. So there's no guarantee that things will stay the same from year to year with those cutworm patches.
Okay, great. And there was one more for you, John. Ingrid had asked if you could comment on some non-pest grasshoppers, and if you have a chance, some early spraying done on hoppers due to numbers.
That's a really good question too. And a few rules that we have among entomologists is that any grasshopper you see that looks really big in April or May won't be a pest species. We do have a few species that overwinter as partially grown nymphs instead of eggs. All our pest species overwinter as eggs, and they won't start hatching until very late May or into June. So by the time they're big, it's going to be later in June or into July even. But we do have a few species, non-pest ones, overwinter as partially grown nymphs. They will be big early on. So if they're large early, not a pest. Any grasshopper that makes noise when it flies, sometimes there's a few species that you'll hear a crackle or crackling when they fly away. They're not pest species. And if they've got colored wings, we've got one that's really common. It's got a the hind wing when they fly is black. It almost looks like a butterfly, black hind wing with a yellow border. That's the Carolina grasshopper. We don't see them invading the crop fields. They usually stick to a lot of your roadside vegetation. Again, another non-pest species. So it is worthwhile getting to know what the pest species are. We've got a fact sheet on our website that goes over that. And again, don't get too panicky if you see a few larger grasshoppers early on. It doesn't mean you're in for an outbreak.
All right, thank you very much, John. And Terry, that's all I have for questions.
Okay, then we're going to wrap up the session for today. I'd like to take the opportunity to thank all of our speakers today. You all did a lot of hard work in preparing for this and meeting the requirements that we set out for you and responding to the questions, and that's very greatly appreciated. I think we did a lot of good work here this morning and a lot of good information was shared. As I say, this was meant to be a primer because it looks like spring's coming quickly. And I'd also like to thank all of the attendees and thank you for sticking with us through this whole morning and for attending. And I hope you got some great value out of this exercise. Thanks very much, everyone. And I wish you all a really good season this crop year.